Dr. Vel Murugan, we can start, I think. Mostly all the participants are joined. Our Vice Chancellor is joined as well, please.
So, Dr. Narendra sir, are you on way?
I just checked with VC sir's PA. I just can get back to you.
Yes, sir, please, sir.
Yeah, VC sir is joined. Dr. Vel Murugan.
Yes, madam. Yes, madam. I'll inform to Narendra.
Yeah, please, you can start.
Okay, so I request the MC team, please.
Yes. Good morning. Day, smile and positive thoughts. A warm welcome to one and all gathered here for the Golden Jubilee Commemorative webinar on exploration of knowledge on science and technological innovations for nation building, organized by Department of Science and Technology and jointly organized by Indian Science Congress Association, Coimbatore Chapter and Kongunadu Arts and Science College, Coimbatore. The most beautiful thing we do in our life for success is praising and worshiping God. Now let us begin this webinar by singing a prayer song to spread the positivity and to glorify the God. I now request ma'am, Assistant Professor in Tamil Department, Kongunadu, Coimbatore, to sing the prayer. Please, ma'am.
भवानी शंकरी पार्वती भवानी शंकरी पार्वती परमदयागरी पार्वती शिवा शिवंकरी पार्वती श्रीपुरवासिनी पार्वती हिमाचल कुमारी पार्वती ईश्वरी शंकरी पार्वती कुमार जननी पार्वती कुंकुमांकिते पार्वती अखिलांडेश्वरी पार्वती आदिपराशक्ति पार्वती अगर्डपूरणी पार्वती आनंदरूपिणी पार्वती भवानी शंकरी पार्वती परमदयागरी पार्वती.
Thank you, ma'am. Nobody can go back and start a new beginning, but anyone can start today and make a new ending. I now humbly welcome and invite Dr. C.A. Vasiki, Madam, convener of Indian Congress Association, Coimbatore Chapter, and Secretary and Director of Kongunadu Arts and Science College, Coimbatore, who is an ideal thinker, fair-minded in evaluation, honest in facing personal biases, persistent in seeking results, to welcome the gathering. Please, ma'am.
Thank you. Pleasant good morning to all the beneficiaries of this eventful webinar, which has been entitled Exploration of Knowledge on Science and Technological Innovations for Nation Building. And before that, I would like to wish everybody a happy Navaratri festival time, and may this pandemic, with the help of all our Shaktis, be eliminated throughout this world. So with that wish, I'm very happy to start this conference today and welcome all the dignitaries. And this has been organized by Indian Science Congress Association, Kolkata, and also the ISCA Coimbatore Chapter and Kongunadu Arts and Science College, Coimbatore, to commemorate the Golden Jubilee of the Department of Science and Technology. As we all know, applied science is very purposeful and determined, and pure science is very playful and curious, and continuously when both applied science and pure science, they stimulate one another and support one another. And today, I think most of our Indian Science Congress committee members, the executive committee members, the president are all basic science people. I'm very glad, and I think our beloved Vice Chancellor is also started his career as a basic science person. So we know how both, when they come hand in hand, it helps in development of this great nation. The nation of the future is, it is very important to protect the freedom of pure science as much it encourages applied science. Today, we have amidst us our Honorable Vice Chancellor, Professor Dr. P. Khaliraj of Bardea University, Coimbatore, who is an eminent biotechnologist and has clearly balanced pure science and applied science in his research. On behalf of Indian Science Congress and Kongunadu Arts and Science College, I wholeheartedly extend a warm welcome to you, sir, and your presence has added flavor to this webinar. My gratitude and a congenial welcome to Dr. Vijayalakshmi Saxena, the General President of ISCA, who is a mammoth support for all the activities organized under the aegis of Indian Science Congress Association. I am also very happy to extend a gracious welcome to all the executive committee members who have joined here. I am very pleased to see all who have joined from each and every corner of our great Bharat. I am very happy to extend a gracious welcome to Professor Dr. R.B. Subramaniam from Sardar Patel University, Gujarat, and Dr. R. Karvembu from NIT Trichy, Dr. N. Ayadurai from CLRI Chennai, and Dr. M. Ramasubramaniam from IIM Uttarakhand. So I'm very pleased to welcome you all, sir. And I also extend a welcome to the proactive organizing team. That is my beloved principal, Dr. Lakshman Sami, and our Dean R&D, Dr. Paul Sami, our faculty members, and our students. I congratulate each and every stakeholder who are the beneficiaries of this webinar and would like to see you all blossom beautifully and contribute to the self-reliant India. I wholeheartedly extend a companionable welcome to you all. India is gloriously marching ahead in this present decade in science and technology, and the historical image of our country globally has gone up to great levels. And let us first thank for our government for introducing, after a very long wait, now we have a big boon, I would say, as an educationist, and all of you will agree with me, is our new educational policy 2020, which has been introduced. And I am sure this is going to foster excellence and in education as well as in research in all spheres. Take it from primary school, a great change is taking place in our education system. And this change was not done just like that, with the chairmanship and the headship of Kasturi Rangan, who was a great scientist, and also a big team of people with who have got the opinion for the past 2 years, have worked like getting opinion from all the stakeholders. In fact, from students to teachers to parents to everybody have contributed in our new educational policy. And I am sure this will rise our standards to a much higher level globally. And India is definitely going to stand outstandingly in the world with knowledge. As we all know, India is a society, it's a knowledge-based society. And let us see some of the important schemes that our government has introduced foster education and research. And one of the important thing which I really liked is our Atmanirbhar Bharat Abhiyan, which is mainly to make India self-reliant. And how do we make it self-reliant? We can become vocal for local goods. And today the self-reliance has, especially during pandemic, India is not suffering like many other countries who are dependent on importing from our basic food to clothing. We are so self-reliant in everything, including we have been supporting other Western countries, the so-called self-reliant in technology, in even our medicines. We have been the first country who have been sent our basic paracetamol throughout the world to Western countries. So I'm very proud, this Atmanirbhar Bharat is definitely going to rise our country to a global standard. And where we are going to enable the other important thing is the Unnat Bharat Abhiyan. I am sure you all know. Now, as you know, the urbanization is causing a great pressure to cities, and Unnat Bharat Abhiyan is going to make our villages very smart villages, and this in turn will make our villages to be self-sufficient and self-reliant. And that is our villages, as our Mahatma Gandhi said, our life and soul of our country. More than 70% of our nation lies in the villages. Our resources are there. Our heart is in the villages of India. I am sure this Unnat Bharat Abhiyan is going to fulfill the dreams of Mahatma Gandhi ji. And now the Ministry of Education gives a lot of importance for Institution Innovation Council for promoting entrepreneurship development among students. Yes, this is important because today, whatever research is there are DST and DBT and various organizations UG is supporting in for research with, but what is happening? How much of is becoming into products? It has been helping the society. So I'm sure this Institution Innovation Council is going to make our visitors and our beloved students very reliant and make them into entrepreneurs and make them into job creator rather than job seekers. So I am very happy. 2 days back, which was our National Innovation Day, and our government has introduced Kapila, which is a program for protecting the intellectual rights. And yes, the IP literacy in India is only 40,000. And for a vast country, and for the amount we spend on research and technology, it is far, far less when compared to even our China, who are a big copycat technology. When you see China, they do not have innovation among them. They are very good copycat people. They copy technology and the ideas from various countries and are good in producing cheap products. So that's not what our aim should be. We should be producing our own indigenous technology and help our society, and not only our society, throughout the world. So with this small note, I'm very happy to welcome all the gathering. And also one more point, our basic structure of Indian Science Congress, always we talk in every meeting, is to improve the scientific temper among our students, our youth, our youngsters, because at the moment people are going more into the loss. There is a great loss of interest in basic science. So people are getting into technological side, but our Indian Science Congress has developed this and giving so many programs to improve the scientific temper among school and college students. So apart from that, my request is that science is a study of knowledge which is made into system, and it depends on analyzing and understanding the facts. So technology is the application of this scientific knowledge. So for any successful economy, we see science technology, engineering, innovation are the real pillars, who are the architects and foundation stones for building a self-reliant, self-sustaining Bharat. The architects who can contribute for this nation building are none other than our beloved students, and the lack of interest can be, I would say, can be alleviated by improving the government scholarships. The central government, state government should increase scholarships for basic science students, so that it will have an attraction, and this will also help us to build the basic knowledge in science, which will help in technological development. So I conclude today by quoting our Sadhguru, who once said, if we transform our population into a trained, focused, balanced, and inspired population, India will be a miracle, which we had during the Vedic period. And this legacy and glory is being revived in current times. I once again welcome everyone and hope you have a great scientific session. Thanks and namaste.
Thank you, ma'am, for your gracious welcome address. Leaders are made by hard work and priceless efforts. I feel much delighted to welcome and invite Dr. Vijayalakshmi Saxena, ma'am, General President, ISCA, Kolkata, who is an eminent and ambitious personality to deliver the presidential address. Please, ma'am.
Very good morning to all the participants of the international webinar, and a happy Navratri to you all. Professor Kaliraj, Vice Chancellor, Bhattacharya University, Coimbatore, Patron Dr. Raviswamy, President, and Dr. Vasuki, Convener, as well as Secretary and Director of this college, Dr. Lakshmana Swamy, Principal of the college, Dr. Madan Shankar, Co-Chair Dr. Paul Swamy, Organizing Secretary Dr. Narendra Kanam, Professor R.V. Subramaniam, Sardar Patel University, Gujarat, Dr. Karvembu, Professor from NIT Tiruchirappalli, Dr. Ayazurai, and Rama Subramaniam from Kashipur, Uttarakhand, members Dr. Dhanan, Dr. Sangeeta, Dr. Kartika, Dr. Rekha, Dr. Velmurugan of KSC. All the participants of the webinar, DST personnel, invited speakers, faculty members, council members, chapter conveners, students, ladies and gentlemen, and media personnel. First of all, I feel privileged to have been granted the opportunity to share my views with the galaxy of eminent scientists. I would like to begin my introductory talk with the famous quote of Victor Vickoff: "Science and technology are only one of the avenues towards reality. Others are equally needed to comprehend the full significance of our existence. Indeed, these other avenues are necessary for the prevention of thoughtless and inhuman abuses of the results of science." I strongly believe that Victor Vickoff's idea of development in science and technology are fundamentally altering the way people live, connect, communicate, and transact, with profound effects on economic development. To promote technically advances, we should invest in quality education for youth and continuous skill training for workers and managers. Science and technology are key drivers to development because technological and scientific revolutions underpin economic advances, improvements in health systems, education, and infrastructure. Indian Science Congress Association is committed to spread the concept of exploration of knowledge on science and technological innovation for national building by pointing out the interdependence of scientific innovation, production, and environmental limits. Today, I am proud that ISCA Control Chapter has organized the webinar on exploration of knowledge on science and technological innovations for nation building to commemorate the Golden Jubilee celebration of Department of Science and Technology. Department of Science and Technology was established in May 1971 with the objective of promoting new areas of science and technology and to play the role of a nodal department for organizing, coordinating, and promoting science and technology activities in the country. The department has major responsibilities for specific projects and programs and the formulation of policies relating to science and technology, like promotion of new areas of science and technology within domestic technology, all measures needed for the promotion of science and technology and their application for the development and security of the nation. Matters relating to institutional science and technology capacity building, including setting up of new institutions and institutional infrastructure.
Hello, I think Lakshmi Madam's connection is gone.
Am I audible? Then I can just start.
Yes, I think so, sir.
Yeah.
Right.
Respected Dr. C.A. Vijayalakshmi, Vijayalakshmi Saxena, Honorable General President of ISCA Kolkata, Honorable Syndicate Member nominated by Chancellor Dr. C.A. Vasuki, Convener of ISCA, and Secretary and Director of Kongunadu Arts and College, Principal Lakshmi Swami, and distinguished speakers, Dr. Bhagavati Subramaniam, Dr. Karvembu, Dr. Ayadari, Dr. Ramasubramaniam, and students of Kongunadu Arts and Science College, and members of ISCA, and student members from the Kongunadu College and other academic institutions. I'm extremely happy to be here with you for inaugurating the
எதுக்கு ரிமூவ் பண்ணிட்டு இருக்கீங்க ஸ்டூடெண்ட்ஸ் if they join let me sorry
Okay, no problem. I'm extremely happy to be here for inaugurating the webinar on exploration of knowledge on science and technological innovations for national building, jointly organized by Commemorative Arts and Science College and ISCA. And the contribution of the faculty members of Commemorative Arts and Science College is enormous. I would like to congratulate them. And as far as the institute, Kongunadu Arts and Science College is concerned, it takes back to 1972 or so, starting from having only pre-university course. Now it has come to the level of international recognition. It is the best model of the institute, instituted by an academician. There's a lot of difference between instituted by various type of people. Dr. R. Swami was the one He has successfully brought up the institute. And the most important thing is he was able to make Dr. Vasiki also to develop the interest into this line for the sustainability. I wish Dr. Vasiki will generate the next generation and motivating them for the several generation, which is an important aspect. And by the time the national education policy is going to be reaping, we might not be very active, but the next generation will take care of that. The most important aspect I would like to appreciate is it's having about 5,000 students, mainly of girls, about 58% are girls, which is an important aspect. And they have a uniform academic excellence in terms of NAC accreditation, A+, consistently for 4 cycle, then Institute got College with Potential for Excellence, then College of Excellence, and having several programs of University Grants Commission, DST, and so on. And the amount of 80 lakhs means allocated by DST under the FIS program is remarkable for the private institutions, and that is because of the hard work of the institute. And I'm admired for this institute is coming at the top 100 ranking of the NRI. I'm very proud to say that in Bharatiya University, affiliated institutions and autonomous, we have about 7 institutions having the ranking less than 100, which is a credit for the university and also for all the affiliated colleges and autonomous institutions. Now, the ISCA, the Science Congress Association, I appreciate the motive of making the teachers research oriented, which is very important. Only a good research worker will be able to be a good teacher. If you are only teaching, you prepare the notes or a PPT, it will be coming for several years. But when you do the research, you are going to read and you keep on publishing in good journals and new information will be passing on to the students. More than that, creating the knowledge of research as well as the motivation of research at the UG level, which is very important. Doing this one research at PhD level is okay, but the student has to be motivated from the undergraduate level itself. Now, what has to be the responsibility of ISCA? Now, according to the new education policy, we are expected to have the GER of more than 50% or 60% in 2030. But Tamil Nadu already we are having the GER of 49.8. Now, what will be our contribution? What is our goal? Well, we are going for that. Then, the most important aspect of looking into the new education policy is school education. Unless we strengthen the school education, we are not going to get a good raw materials for our higher education. So what we have to do? The most important thing is strengthening the capabilities of the school teachers. Now, who is going to strengthen them? It's not the school. It is academic higher education institutions and also organizations like ISCA. Last week we had the program of AICTE, Faculty Development Program for CBSE teachers. I was very much, impressed by the activity of AICTE. Why you are targeting the school teachers? Because they are the one who is going to give the best possible students for your higher education. Unless we do well for the undergraduate program, we can't get good students for the PG program, then for the PhD program. It starts from the school. So what this organization can be done? Now you have to motivate the teachers of the higher education in various academic institutions. And government institutions is one side. And we can always take help of some of the institute like Kongunadu. Make these teachers to give the orientation to the school teachers, just like what AICTE has done. There are many programs of AICTE also. and how they have to motivate the students for the higher education. And today, the career guidance is also very, very important. 95% of the school students, they are taking up the career only based on the advice of the school teachers and also to some extent by the parents. So how many students are taking the right path? Are they taking the areas of their interest, or they are telling just because of the teachers are telling engineering is good, or it is medicine is good, or law is good? So you have to go for the psychometric analysis. Psychometric analysis of the school children are very, very important. We have done, our career guidance, Dr. Vimala has done psychometric analysis in the Saura school and few other schools in the nearby area of Bardea University. We could see that 90% of the students are depending on the teachers and parents. The parents are going to tell, oh, you go for medicine or you go for engineering. That is a tendency of the parents because of their anxiety. Even I am also example for that. My daughter was interested in biotechnology, but I asked her to go for electronics and communication engineering. I thought that will be better, but even now she is accusing me. I could see that. And after she completed ECE from Anna University and MBA from Bangalore University, she has completed Diploma in Medical Lab Technology from Stella Maris College, and she is taking care of my wife family business of diagnostic industry with, laboratory, CT scan and all. So that means her mind is with science. A poor parent like me, I was telling Anna University EC is best one. If you ask my daughter, she will accuse Anna University for anything. Because I have been given the education in the field which I was not interested. So let this association promote the motivation of our higher education teachers to get into the schools. At least let us adapt. You adapt a few villages, adapt some schools, and make it as an example. Go for psychometric analysis and give the certificate. We give the certificate to the school children in which particular field that he will be doing well. And when the parents are forcing him, he will be showing that certificate on the psychometric analysis of the higher education institution. I am suitable to go as an engineer or a lawyer or in this one medical person. So that will be giving the way for that. So that should be the responsibility of organization like ISCA in collaboration with the higher education institution. Of course, I need not have to tell about DST. DST is the only one organization. They don't see the hierarchy of any approval. If you are going from, now only you are having some funds allocated by UGC for the private institutions. Sometimes they'll give 12B status without funding. What is the use of that? And if you are private, you are not eligible for that, but DST is giving for everyone. It all depends on what kind of project you are having. A lot of Prime Minister's ideas, schemes and all, they are all promoting the academic institutions. And I'm very happy to inform you that, Bardea University, because, many members are from outside, consistently for the last 5 years, we are having the NIRF ranking, the university category of 13 and overall category of 21. And this year, this is the first year we made an attempt, because none of them made an attempt thinking that we might not be getting world university ranking. I told, if you are having consistently a NIRF ranking, you just hit into that. So in Times World University ranking, we got Indian ranking 19 and global ranking 8.8. This is a contribution of my previous vice chancellors, previous HODs, previous faculty. So I should go for bringing down to 500 to 600. That is my motive. I'm working for that. And even in Shanghai ranking, Shanghai world ranking, we were able to break the barrier of 1,000. We have gone in between 900 to 1,000. And I have to bring it back down at least to 700 to 800, something like that. And this time, with all this COVID pandemic, this one time, gave a lot of time to me. No need of going to Kongunadu Arts College for inauguration. No need of going to any institute for any seminar and all. Sitting at university, and we were able to introduce part of the national education policy. National education policy might have been introduced recently, but the draft has come a year back. And our Chancellor Vision 2030, even before the pandemic, they wanted to go for Chancellor Vision 2030, which was meant for incorporating Industry 4 and 5. So we incorporated the vocational courses, job-oriented certificate courses, MOOCs courses, add-on courses, and industry participation in all the syllabus. Not only our university syllabus, UG and PG syllabus of our affiliated colleges. And we have gone for the outcome-based syllabus. And we are introducing this particular academic year. And we are able to conduct this online evaluation very nicely. I'm very happy to say all the autonomous institutions, they have done online exam and evaluation just like our regular examination through Google Meet. And I know they have to use their university site for downloading the question paper and uploading the regular answer paper. And we have the bell-shaped curve of our results. Yesterday only we declared our UG result, PG result will be ready in 2 days. So we have to go for adapting the modern technology, industries 4 and 5. So this year, the industries 4 components like big data analytics, Internet of Things, artificial intelligence, mobile app, augmented reality in all the subjects, including your economics, mathematics, and linguistics and English. How we are going for the artificial intelligence and augmented reality for your linguistics. How are you going to use it for the translation? So we have to go a long way within a short time. So by the year 2023, our students will be entirely different what you are having it now. So the type of job that you are going to have is different from the type of job that you are having it now. So it is a responsibility of organization like your Indian Science Congress Association and institute like Kongunadu Arts College and other academic institutions and higher education governing bodies like universities, and Bharatiya University is there. And I am promoting the NRF ranking for all the institutions. You try, you find out where you are. At least you will be able to try to go towards the forward direction. So I will give all cooperation for all the institutions, including the autonomous institutions. And this program will be highly successful. I know many of the stalwarts are here. They're all, promoting the education for the college teachers. It's very important. There was a time research means only it has to be done in the universities. Now the things got changed. I am seeing a journal in Kongunadu College, and a lot of good papers are coming. It's a referred journal, and good number of PhD scholars, and almost all the faculty members are holding PhD. And the situation has to improve. It's not only the university. The research has to happen at the UG level and PG level in colleges as well as the university. Unless you give a good UG students with research attitude, you will not be able to have good PG students for you or for our university. So I wish the program all success. Thank you very much, Dr. Vasvi Madam. After you become the syndicate member, we could not make. And even our syndicate meeting, it was only through online. Looking forward to meet you soon.
Thank you, sir. Thank you. Thank you for a very innovative and you gave excellent ideas, and definitely we will take it all up. And thank you for your valuable time you have given us to give this address. I'm very keen to meet you as well. Thank you, sir.
Thank you, madam. Now we can request Vijayalakshmi madam to continue her talk.
Madam, Vijayalakshmi madam, I think you got Vijayalakshmi madam, now you can please proceed. I think you got disconnected. And madam, if you don't mind, please can you proceed?
Or you leave it. Whatever you You please
Yeah,
continue.
please, madam, you proceed,
I
please.
think you
Because
conclude. Because,
Okay.
not conclude.
you're completely, we couldn't hear you. So our Vice Chancellor utilized the time.
But 5 or 10 minutes you have already listened, no? Or
Yes,
that's-
yes, yes. But please continue, madam, because you got
Okay.
disconnected in the middle.
Okay.
Thank you, madam. Thank you.
Which may disconnect you. The role of science and technology in our future. Indian society may be broadly stated as to ensure national security and social stability, to sustain the growth of the national economy and to improve its efficiency, to prepare for a smooth transition to an information society, to improve the quality of life, to create a new culture suitable for a new society. There are many success stories of DST, like women empowerment of Himachal women through post-harvest technology, rural women technology, Park Satara Maharashtra, NBIRT Vision for Tomorrow, FIRST program, SEED program, Initiative on Enhancement and Securing Women and Child. The Department of Science and Technology has launched a scheme named Visiting Advanced Joint Research Faculty Vajra, in a bid to bring together the Indian scientists abroad and India-based researchers for conducting joint researches in India. The National Education Policy 2020, which was approved by the Union Cabinet of India on 29 July 2020, outlines the vision of India's new education system. The new policy replaces the previous national policy on education 1986. The rich heritage of ancient and eternal Indian knowledge and the thought has been a guiding light for this policy. The Department of Science and Technology have initiated the formulation of the new National Science, Technology and Innovation Policy, STEP 2020. It will be the fifth step of India and is being formulated at a crucial juncture where India and the world are tracking the COVID-19. It will integrate the lessons of the pandemic, including the building of an Atmanirbhar Bharat or self-reliant India vision of our Prime Minister Shri Narendra Modi by leveraging India's strength in research and development, science and technology, workforce and institutions, huge markets, demographic dividend, diversity and data. Dr. Harshvardhan, the Union Minister for Science and Technology, Earth Sciences and Health and Family Welfare releases white paper on focused on intervention for Make in India post-COVID-19 with the new mantra, local solution to global challenges, policy and technology imperative. Professor Ashutosh Sharma, Secretary DST, said the white paper by Effect Technology Information Forecasting and Assessment Council presents a compelling map of high priority sectors, technologies, and strategies to fuel growth in the time of COVID-19. I'm sure new innovations which have been won in science in all different fields, the credit goes to DST, like agriculture, space research, defense research, atomic research are actually the seed sown by the DST. We are citizens of the nation and who hold equal responsibility for the growth should equip our youth with all possible facilities for their research first and support and motivate them, as the future of a nation is in their hands and they could get our nation to more advanced levels than what is today. I feel privileged in reproducing an excerpt from the very motivational and accelerating speech of our most devoted Prime Minister he had delivered in connection with making India self-reliant. We walk together, we move together, we think together, we resolve together, and together we take this country forward. Thank you all.
Thank you,
so
madam.
much, ma'am. The world is round and the place you see may look like an end, but it may also be the beginning. I take this opportunity to invite Dr. M. Lakshmanaswamy Sir, Principal, Kongunadu Arts and Science College, Coimbatore, to propose the vote of thanks. Please, sir.
Good morning, one and all present here. With immense pleasure. First, I would like to thank our Secretary and Syndicate Member of Bharati University, Dr. C. Vasuki Madam, Convener, ISCA Coimbatore Chapter, for her welcome address. And last 6 months, Madam was highlighting and addressing all the state level and national level meeting for the importance of the new education policy. I'm very happy to share. The Madam also highlighted the various schemes by our central government, the Atma Nirbhar Bharat and UBA, Unnat Bharat Abhiyan. And last month, our college has received rupees 50,000 from the central government for the mushroom cultivation in our NSS adopted villages. And DST, DBT, UGC, DRDO, and the Department of Environment. And Kabila, there's a new project launched by our the Innovation Institution Innovation Council, that Kabila, for intellectual property right. At this juncture, I would like to inform that there's one good news, that is 15th October, our college has awarded 5-star status by Institute Innovation Council for our innovation based on our academic activities. So this is because of the effort of our secretary, staff members, and the stakeholders. Thank you for your valuable content in the cause of the higher education. Then coming to the next, I want to thank Dr. Vijayalakshmi Saxena Madam, General President, Indian Science Congress Association, and Kolkata. So we want to participate either Kolkata or Coimbatore chapter or Kolkata chapter. The role of and the contribution of Professor Saxena and Dr. Vijayalakshmi Saxena are enormous. And Madam also, in her speech, he highlighted that science and technology, the role of science and technology activities for our national building, mainly the rural women technology and the new innovation, the connection with the new education policy. Thank you, Madam, for your contribution for the development of the Indian Science Congress Association in Kolkata and also our in Coimbatore chapter. Next, I want to thank our respected Professor Dr. P. Kaldiraj Sir, Vice Chancellor Bharati University. Sir, I'm on behalf of the management, definitely, sir, I'm more interested to thank you, sir. So first of all, you have highlighted the significance of our college, that the total number of strength 5,000 and the 58 point women, it shows the women empowerment, 58% of girl students, and the 4th cycle NAC accreditation, College of Excellence and Potential for Excellence, UGC and the FIST program around more than 80 to 90 lakhs for the FIST program, and the NIRF rank 70th. And for all the members that I want to mention, that the long vision of our Vice Chancellor, the last 1 month, 1 week, our Professor Kaldiraj Sir is conducting a various meeting and orientation program for all the experts and the college professors, how to conduct the online exams due to the pandemic situation. Sir, we are all trained in our college, it's an autonomous college. We are already conducting the examination for final year, and also the 8th of October, we have published the results for the final year students, and we are expecting the university result also. Then only after that, we want to continue our PG admission. And again, our professor, once in a week, he has given training to all the teaching and non-teaching of all the affiliated and concerned colleges. And it shows the long vision. And Professor highlighted that what are the responsibilities of the ISCA, and mainly the gross enrollment ratio 60 to 70% in 2020 in the new education policy, and to strengthen the school education and to strengthen the UG. Once you can, we will fill the UG, we have no problem for admission to PG. Once if you strengthen the UG admission and the PG admission, there is no problem for the research admission. Thank you, sir. And again, he stressed the science in the school level and world ranking. Really appreciate, sir, for your university staff members and the former Vice Chancellor and your enormous contribution for India ranking the 19th and the Shanghai ranking. On behalf of the management, I take this opportunity to thank you for your valuable contribution to develop our university in the world level. and online and offline, you are training us more useful. Thank you, sir. And I'm more indebted to thank our Professor Gangadhar sir and Manoj Chakrabarti and our resource patients, Dr. R. Bhagavati Subramaniam sir and Dr. R. Karvembu. Karvembu professor was already working in our chemistry department self-finance course. Dr. N. Ayathore sir and Dr. M. Ramasubramanian sir, faculty members, participants, state level and national level, research scholars, and my dear student friends, and the organizers. Co-chairperson Dr. S. Balusamy sir, Dean R&D, Coordinator Dr. S. L. Madan Shankar, Dean Academics, Organizing Secretary Dr. Narendra Kannan, Department of Biochemistry, Dr. M. Dhanam Madam, Assistant Professor and Head, Department of Physics, Dr. V. Sang, Assistant Professor and Head, Department of Chemistry, Dr. Karthika and Dr. R. Department of Botany, Dr. K. Velmurugan and Dr. C. Sobana, Assistant Professor, Department of Zoology, and other the technical staff members on behalf of the management. Once again, I thank you all for the opportunity given to me. I wish the program a grand success. Thank you.
Thank you, sir. We have few announcements. We have 4 technical sessions under 4 different topics comprising
Thank you, sir.
45 minutes.
Thank you. Thank you.
At the end of each session, we will have 5 minutes time for discussion. The participants can either ask directly or post their queries in chat box. We are now moving on forward for the technical sessions. To be inspired is great, but to be an inspiration is honor. I am much privileged and honored to introduce and invite our guest speaker, Dr. R.B. Subramaniam sir, Professor, BRD School of Biosciences, Sardar Patel University, Gujarat. Sir has completed his Masters in Botany in Madurai Kamaraj University, PhD in Botany in Sardar Patel University, and postdoctoral research in IISc Bangalore in plant biotechnology. His major research interest areas are plant and fungal biotechnology. He teaches instrumentation, genetic engineering, and medical botany to postgraduate students. Sir worked as a lecturer, reader, and professor in Sardar Patel University, and now working in the Center for Interdisciplinary Studies in Science and Technology as a director from June 2018 in the same university. Sir has been elected as a fellow of Gujarat Science Academy in the year 2007 and National Associateship Award by DBT in the year 1997 to 1998. Sir has reviewed and is reviewing research papers for various international reputed journals like Journal of Experimental Botany, Journal of Biosciences, and Indian Journal of Biotechnology. Sir has visited countries like South Korea, Hong Kong, and Indonesia. Sir has published 1 book and 80 research papers, and he is having 1 patent. Sir served as an organizing secretary for various hands-on training program and conferences and has completed 2 major projects funded by CEIB in New Delhi in 2010 to 2016 and BASF in 2014 to 2015. And now I request you, sir, to deliver your valuable invited talk on the topic strobilurin and the role in plant defense and development. Please, sir.
Yeah. Thank you so much for a very elaborate and nice introduction. First of all, I would like to thank the organizers for giving me this opportunity. In fact, I have been associated with Kongunadu Science College for a long time. One of my close friends, Dr. Armugaswamy, was working there for a long time. In fact, he did his PhD from my department. So I have delivered many lectures in the same college, and it's a pleasure to be associated with this management. Recently, also a few years back, Dr. Palswamy invited me, and I delivered a lecture in one of their conferences. So once again, I thank all the organizers, Dr. Vasuki Madam, Dr. Palswamy, and Dr. Narendra Kannan for giving me this opportunity to discuss a part of my research work which I will try to simplify because I understand that the audience have come from various disciplines of science. So if I get into hardcore molecular biology, it may be a bit difficult for them to understand. So wherever possible, try to simplify, but then, research is research. We can't go beyond a level of simplification. So with that brief introduction, let me start my presentation. So once again, a very good morning to all. I have, I think, 45 minutes of time. We are running a bit late, so I'll try to complete my talk within that time period. The topic that I'm going to talk today is on a very interesting molecule that we had come across in the year 2014. And we have been working on this molecule for the last 6 years. And we are very excited about this molecule, and we are continuing to work on this molecule, and we look forward to some great work on this molecule. This molecule is known as strobilurin. It's a group of compound, and this compound has interesting effect on plants. So in today's talk, I will be discussing about the effect of this group of substances on the physiology, the yield of plants, with an idea that this compound could become one of the smart molecules which can be used in future for plant development as well as plant growth. Now, if you look at the annual grain production in India, as I got from the FAO, in the year 2017-18, we have produced a total of 284.83 million tons of cereals. And in the year 2030, we would be requiring around 313 million tons of grains, which is a shortfall of around 30 million tons. If you look at percentage-wise, it is just 10% of increase in the next 10 years. But 10% increase of cereal production alone in the next 10 years is next to impossible, because we are losing a lot of agricultural land, and farmers are also shifting to cultivating other crops rather than cereals because they are not getting much money in cereals. In fact, farmers are recently shifting towards horticultural crops, and the area of cultivation for horticultural crop is greatly increasing. Thereby, the available land for cereal production will decline in the coming years. So now, if this is a situation, what we should do? What are the options before us? We have several options, but I have mentioned 3 important options that we can think of. The first option is to go for what are known as smart plants. Now, what are these smart plants? Plants which can adapt themselves to various climatic conditions. Climate sensitive crops, or we can say climate ready crops. Crops which can grow in varying climates. For example, if it is cold or hot, if it is raining or there is a drought, these plants will continue to grow. Such plants are called smart plants. It is quite possible to achieve production of smart plants through genetic engineering. The next option before us is to improve on the fertilizers that we are currently using. We can produce what are called smart fertilizers. In my next slide, I will present a few facts about these smart fertilizers. In fact, there is a very recent publication on smart fertilizers which came in last month, 2020, which reviews about the development of smart fertilizers for crop production. The third option before us is to use multifunctional compounds. In fact, my today's talk will mainly focus upon these multifunctional compounds. What are these multifunctional compounds? Let me describe very briefly. We are using several compounds in the form of pesticides or herbicides or fungicides or in the form of fertilizers or plant growth enhancing compounds. If a compound can do more than one function, for example, if a pesticide can also promote plant growth, or if a plant growth promoter can also act as a fungicide, then such substances are known as multifunctional compounds. The advantage of using such a multifunctional compound is that the farmer can save a lot of money, and instead of using different compounds at different time periods, by using a single substance, he can achieve both control of diseases as well as enhancement of plant growth. The strobilurin that I mentioned in my talk, in the beginning of my talk, is such a multifunctional compound which is capable of acting both as a fungicide as well as a plant growth promoter. Now, what are these smart fertilizers? I told you that I will describe briefly what are these smart fertilizers. I think all of you must be knowing about normal fertilizers. Major fertilizers that are used are NPK, nitrogen, potassium, and phosphate. These are the major elements which are required for plants and their growth. But smart fertilizers are those which are coated with biopolymers, which are biodegradable. And once you put them in the soil, they will stay in the soil for a long time and continue to leach the fertilizer coated inside to the plants. This way, the leaching of the fertilizers from the soil during rains or irrigation and the wastage of fertilizers can be avoided. And by just putting a little quantity of fertilizer exactly around the plants, the farmer can also save a lot of money. A number of biopolymer coated slow release fertilizers have already been developed. And if you do the paper, the title and the citation of which I have shown at the bottom of my slide, you will understand the categories of biopolymer coated slow release fertilizers that have already been developed by various countries, including China and USA. They are going on a big way for developing this biopolymer coated slow release fertilizers. Then there are fertilizers which are tailor-made. For example, you can develop a specific mixture of fertilizers which are meant only for tomato, or a mixture of fertilizers which are meant only for potato. These are known as tailor-made fertilizers, because every crop requires a different proportion of these nutrients. By making this mixture and supplying them to the plant, you can maximize the growth of the plants. The third and the most exciting category of smart fertilizers are known as targetable fertilizers. Little research has been done, but there is a very big scope in this area. And targetable fertilizers are those which can be targeted directly to the plant root. The overall principle behind the targetable fertilizer is that there are certain chemoattractants or chemosensors which are attracted by the plant root system or the rhizospheric system. If you can integrate such chemosensors with your fertilizers, the fertilizer molecule will be attracted by the plant root, and thus the fertilizer will directly reach the plant root instead of getting wasted in the soil. Now, these are some examples of smart fertilizers, and more detail you can find in the paper that I have mentioned at the bottom of this slide. Now, coming to the topic of today's discussion, strobilurins. Let me first describe to you what are these strobilurins and what is their mechanism of action. Once we understand that, we will get into their uses and advantages. Strobilurins are in fact one of the newest classes of fungicides to enter into the market. Please remember, these are already commercially available substances. This is not something that we have discovered in our lab or we have invented. This is a commercial product. It is one of the newest class of fungicides. It came into the market in the year 2011. Discovered by many companies which are involved in plant growth and development, like BASF, Syngenta, and all. Originally, this compound was isolated from a wood rot fungus known as Strobilurus tenacellus. It's a Basidiomycetes fungus. Scientists, when they were looking at the growth of this fungus, they found that this fungus will not allow any other fungus to grow in its vicinity. Therefore, they thought that there is something in this fungus which inhibits the growth of other fungus. And when they isolated, they found that the compound is a group of substances called strobilurin. Now, interestingly, strobilurin compounds are registered as the lowest risk fungicides by the Environmental Protection Agency of USA. This is one big advantage of strobilurin. They are non-toxic to humans. And another added advantage is that they can control all the 4 classes of pathogens: basidiomycetes, ascomycetes, zygomycetes, and so on. In fact, they control an unusually wide variety of fungal diseases, including water molds, downy mildews, powdery mildews, leaf spots, rusts, fruit rots, and blights. Around 82 different diseases can be controlled by this single compound. So it has a very wide range of action on the fungus. That is another advantage. Now, this is a slide that simply shows you the structure of various strobilurins. These are all natural substances: azoxystrobin, picostrobin, chrysoxym-methyl, trifloxystrobin, milominostrophin, and pyroclostrobin. We used in our study pyroclostrobin. That is why I put it in a box. Pyroclostrobin is a kind of strobilurin compound, which was also originally isolated from Strobilurus tenacellus. But all these compounds, azoxystrobin or picostrobin or pyroclostrobin, these are all photolabile substances, in the sense that when we expose them to sunlight, they will get degraded, they will be broken down, and therefore they cannot act on the fungus. So the first job was to modify this compound, which I will show a little later. Now let us look at the mechanism of these strobilurins, how they act. Basically, the strobilurin compounds have a great affinity to waxy substances, non-polar substances. Now you all know that the plant leaves contain a waxy coating called cuticle, and therefore this particular compound has a natural affinity towards cuticle, and then it actually binds to the cuticle. Once it binds to the cuticle, it enters, it diffuses the leaf, and it can cross the entire lamina and reach the cuticle on the other side because of its affinity towards the non-polar cuticle around the leaf. So it is not acting only on one surface, it can act on both the surfaces, bilaminar, and it can act systemic because it has already entered into the plant cell. So it can act on the surface, it can act systemically, it can act on both surfaces of the leaf. That is the beauty of this compound. Unlike any other fungicide which acts only on the spot where it is sprayed or only systemically, this can act at local as well as systemic levels. Now, what is the mechanism? How this compound inhibits the growth of the fungus? Basically, strobilurins inhibit the mitochondrial respiration. Now, you all know that the electron transport that happens in mitochondria is the key step in the ATP formation. Actually, what these strobilurins do is they block the transport of electrons from the cytochrome B to cytochrome C. This transfer is affected from complex 3, and because of the blockage of this transfer of electrons between complex 3 and 4, the ATP synthesis is blocked. Because of the lack of energy, because ATP is required for various cellular activities, the fungus will die. Now, you can ask me, if this is the case with the fungus, won't the ATP production of the leaf cells be affected? Yes, the leaf cell ATP synthesis is also affected, but to a very, very low level in plants, because the threshold level at which it is toxic to fungus is very high, and at that threshold concentration, the compound is not toxic to plant cells. It is slightly toxic in the sense, to some extent, the ATP synthesis is affected, but it is a beneficial activity, which I will describe a little later. Actually, it's a blessing in disguise for the plants, because the plants could do more photosynthesis. Okay, so that's the mechanism. Now, this is what I have been talking about earlier, the modification of strobilurins. You can see that the ring structure is highly photolabile, and therefore, when they are exposed to sunlight, they get broken down. Chemically, they are modified and they are made non-photolabile and stable. And such chemically synthesized strobilurins are being used as a fungicide today, not the naturally obtained ones. Now, a number of studies have been already done because this compound came in 2011. And when farmers started using this compound on soybean, actually this compound was first prescribed for some fungal disease in soybean in the Latin American countries. When the farmers started using this compound on soybean, they found that the leaves of the soybean plant on which the compound was sprayed stayed green for a longer time compared to those plants which have not been sprayed with this substance. Though there was some delay in the yield, they got more yield at the end of the harvest compared to those plants which have been not treated with this fungicide. So originally the compound was actually used as a fungicide only, but accidentally it was discovered that this compound has also other physiological effects on plants, and then it can be beneficially used even on normal plants, plants which are not affected by any fungal pathogen, to improve the yield. So that is how this compound was actually discovered. It was serendipity, basically. Farmers observed the stay green effect on soybean, reported it to their chemist, and the chemist reported it back to the company, that is BASF, and they then started investigating. In fact, I was part of this investigation in India. I got the project from BASF for continuously 4 years to investigate upon this compound. So in this slide, see that when we use Jubilee, has all including ACE, and here you can see a picture showing a set of plants which are sprayed with fluorine, and a set of plants which are sprayed in the field. See that the plants which have been sprayed with this toluene compounds are greener compared to the plants which are not sprayed with the toluene. Or untreated plants are already drying up. They have finished their life cycle, while the treated plants continue to remain green because their chlorophyll can stay intact for a longer time. Senescence has been delayed, and therefore it will produce more of food by photosynthesis, therefore more of yield in the plant. So we started investigating the effect of this pyroclostrobin in detail. Thought that we should directly go for a field trial because many we carry experiment in the greenhouse pot, but these studies can be extrapolated to field level studies, because what happens in the field cannot be exactly replicated inside a greenhouse or on a pot. So we thought it is better that we directly go to a field and start experimenting on this pyroclostrobin, which is a strobilurine compound. Here you can see the picture showing the field trial. We took a 1 acre land adjoining to our university from a farmer on lease, and we started cultivating tomato with the help of the same farmer. All the farmer practices were followed, in the sense that whatever the farmer will normally do, that was allowed to be done. We did not control any activity. We in fact wanted to see what is the effect of this compound under farmer practices, not under controlled conditions. So 4 treatments were given, 4 sprays were given at 3 milligram per liter concentration. We required around 16 pumps of 50 liters each for the 4 sprays. The first spray was given 40 days after sowing the seeds of tomato. The second spray was given 60 days after sowing. The third spray was given 20 days after that. And the last spray was given just before harvesting, that is after 100 days. In 120 days, the fruit should be ready for harvesting. We collected 40 days old small plants, and we simply looked at their anatomy. Basically, I'm a botanist, though, due to the pressure of times, I have shifted to biotechnology. I'm a basic botanist. So I looked at the anatomy of these plants, and to our surprise, we found that the treated plants had a very elaborate vascular system, especially the xylem vessels, as you can see from here. See the xylem vessels of the control plants. You can see that the cell walls are very thin, not very darkly stained, not much of lignification. But if you compare the same thing here in the treated plants, you can see a very well developed vasculature, which indicates that the plant is already adapting itself to uptake more water and nutrients from the soil compared to the one which has not been treated. This is what this compound is doing. Very interesting physiological effects. And then we studied the effect of this pyroclostrobin on the various biochemical parameters of tomato plant. We first checked the chlorophyll content, and as we expected, the chlorophyll content of the treated, which is shown in dark green color, was always higher in all the 4 treatments. Interestingly, nitrate reductase activity, which converts nitrate to nitrite, and a very important part of nitrogen metabolism, was enhanced. This has already been reported. This is nothing new that we have discovered. We only confirmed what already has been reported. And we found, of course, the lycopene, that is a pigment that is found in tomato fruits, to be enhanced. And ethylene content. Ethylene is a hormone that promotes senescence. So if ethylene is produced in your plant, the leaves will fall early, photosynthesis would stop. But this particular compound suppresses ethylene formation, therefore the leaves stay green and stay on the plant for a longer time. And because of that, photosynthesis is increased, more food is stored, and more productivity occurs. You can see the treated plants and their ethylene level, it is much, much lower compared to the ethylene of control plants, which are not treated. Now, looking at the yield data, which I will show in the next slide. But before that, we also looked the number of fruit bunches, the number of fruits, the number of fruit bunches, and the total number of fruits. You can see a great change in the number of fruits, total number of fruits in the treated plants against 19.53 average number of fruits in a plant. It increased almost to 87.4, an increase of more than 4 times. It's a great increase. It was unbelievable. In fact, we did the field trial 2 times, 2 years consecutively, and we got the same results. So this is a data that shows, the changes in the number of fruits per bunch, number of fruit bunches, and total number of fruits. We had just 3.1 fruits per bunch. In the control, in treated, it went up to 4.6, 6.3, and 19.9, 19.5, and 87.4. So you can see that it enhances all the phenological characters of the plant. Fruit length increased. We measured by a vernier caliper, meter slightly, weight increased. Look at the total yield. This is a very, very important data. Total yield of tomato plants under field conditions per acre. Please remember, we repeated this twice. Per acre, the yield went up 7 times. Against 100 grams for 100 fruits, it went up to 712 kilograms on treatment. Not only that, the number of locules, or the chambers that is present, was also increased from 2 to 3 to 4 on treatment. The fruits got bigger with more chambers, more pigment, and bigger in size. Wonderful effects of this compound on tomato. So this is all known. In fact, of course, in tomato, nobody studied. We did it for the first time, but people have studied it on rice. They have studied it on maize. They have also studied it on various other cereals, like soybean and other oilseed crops also. And the same thing has been reported, that it enhances the yield, but not to the same level that we had reported in tomato. Okay, so we were now interested. Being a botanist turned into a molecular biologist, I was very much intrigued, and I wanted to know what is happening at the molecular level. No study was done till then at the molecular level to understand what this particular compound is doing inside a plant cell, how it enhances all these activities. Because, sometimes it becomes unbelievable, that kind of effect this compound shows. So a fungicide, which is originally meant for killing only pathogens, can it do all these activities? Is it true? If so, what is happening inside the molecular level? That is what we wanted to see. There are 3 different approaches to study the action of any particular compound on a plant. One is called subtractive hybridization. This technique is a bit old. though very reliable, but it gives very limited information. The second approach is northern blotting, again very limited and very laborious. We adapted a third approach that is called whole transcriptome analysis. I think biologists know very well that when a gene is to be expressed in a plant or a protein is to be synthesized, the gene is first transcribed into an mRNA. So if you study the mRNA population, you actually get an idea of what kind of genes are being expressed. Simply, if you study the number of genes that is found in a particular cell, that doesn't tell you actually whether proteins are going to be produced or not, because genes may be there, proteins may not be produced. But if you find a transcriptome, an mRNA, then it tells you that this protein is going to be produced. So we wanted to adapt this technique of whole transcriptome analysis. A whole transcriptome analysis is a technique wherein we look at all the mRNAs that are expressed in a particular organism under a particular set of experimental conditions. For example, in this case, we took the total mRNA expressed in a control leaf of tomato plant after 4 sprays of pyroclostrobin, and I mean in a control plant where there was no spray, and a plant which was sprayed 4 times with pyroclostrobin at an interval of 20 days each. So that is the comparison that we did. This is the workflow. We isolated the RNA by using a specific method that we have developed in our lab using a reagent called trizol. We didn't use any kit. Then we prepared a cDNA library by using a kit from Stratagene. Then we went for whole transcriptome sequencing by using Illumina method of sequencing. Now, this picture shows you the RNA sample that we obtained. You can see the quality. We got very good quality of mRNA. In fact, this is of 28S rRNA. mRNA cannot be seen on the gels. But this band tells you that your RNA is good and it's suitable for further analysis. Then we also checked the concentration as well as the yield as well as the purity of our RNA sample. This is the control RNA. We got a concentration of 490 nanogram per microliter, which is good enough. So the yield was around 12.2 microgram total yield. The ratio of 260 by 280, as many of you must be knowing, if you want to know the protein contamination, we take an absorbance at 260 by 280. 280 is for proteins, 260 is for RNA. The ratio should be more than 1.8. If it is below 1.8, your RNA quality is bad. If it is above 1.8, your RNA quality is very good. So we got a ratio of 1.88 as well as 1.84 for the treated as well as the control RNA samples. So this RNA sample was then subjected for whole genome sequencing. We got 2 reads. We read the mRNA twice in the same machine to confirm that the reading that we got is perfect. The total number of reads that we got was something like 18 lakhs 30,000, 1 crore 83 lakhs 4,924 for the control, and 2 crore 19 lakh 48,178 for the control. So adding both, we got a total data set of around 2.64 giga base pairs. 1 kilo base pair is 1,000, 1 mega base pair is 10 raised to 6. 1 giga base pair is equal to 10 raised to 9 base pairs. So we got 2.64 into 10 raised to 9, as well as 3.21 into 10 raised to 9 base pairs sequenced. So this is much higher than the total amount of RNA, and that is how it should be done, because you don't miss anything. Sorry. Then we did a bioinformatic analysis that is called a gene ontology analysis. Let me explain what is this gene ontology analysis briefly, so that you understand what it implies. Gene ontology analysis is a bioinformatic initiative which unifies the represented gene and gene products of all the species. So if you say metabolic processes in plants, across the plants, whatever genes are involved in metabolic processes, all these genes are grouped together. So that is what we call gene ontology. Basically, it focuses on the function of genes and gene productivity. It's a representative of something that we already know. So if we have annotated the gene function, then only we can do this gene ontology. Fortunately, tomato genome has been sequenced and it has been annotated, so we could do this gene ontology analysis for our RNA data. You can see that at least 3 important processes are highly induced. You can see the bar graph here: metabolic processes, cellular processes, and biological regulation. These 3 processes are highly enhanced compared to other processes. Similarly, binding, catalytic activity, they were also enhanced, and cell and organelle production was also enhanced. So all this indicate a positive effect of our compound on the tomato plant. Now, having the data from the sequence of mRNAs that we have isolated through transcriptome analysis, we wanted to find out what kind of metabolic pathways are getting induced in the treated plants compared to the control plants. Now, to do that, we have to use an analysis called KEGG analysis. KEGG stands for Kyoto Encyclopedia of Genes and Genomes. If you run your data set in the KEGG program, which is freely available, you will come to know what kind of metabolic activities are going on inside your plant cell due to the treatment of pyroclostrobin. We found more than 10 metabolic pathways being induced by the pyroclostrobin. I have shortlisted only 4 which are very important. I have not shown the remaining biosynthetic pathways due to the restriction of time. I've given only 45 minutes. So in 45 minutes, it would be impossible for me to discuss all the metabolic pathways. So we found that the diterpene pathway, biosynthetic pathway, is enhanced. Look at this complex network of metabolic pathway. It looks like an electronic circuit. That is how the metabolic pathways actually work. None of the metabolic pathways run independently. They are all interconnected to each other. And here you can see that the entire diterpene biosynthesis is induced. All those green boxes indicate inducible genes, or genes which are induced by our compound. You can see the green box all along till the end of this diterpene. One of the important diterpene that was synthesized or induced by the pyroclostrobin was gibberellic acid. Now, all the botany students would know very well that gibberellic acid is a hormone which is important for the internode elongation. The plants get taller only because of the expression of this gibberellic acid as a hormone. If the plant has to grow, it must have gibberellic acid. Interestingly, our compound enhances this gibberellic acid biosynthesis. The compound also interestingly enhanced the synthesis of a very important protein called rubisco. Rubisco stands for the protein ribulose bisphosphate carboxylase, the protein that is responsible for photosynthesis. It is found inside the chlorophyll, inside the chloroplast, and because of this protein only, atmospheric carbon is fixed. In other words, if rubisco is overexpressed, there is more photosynthesis, and that is what we found in our study, that the rubisco is being overexpressed in this metabolic pathway. The third important gene that we found to be overexpressed was nitrate reductase. I have already mentioned about this nitrate reductase in one of my earlier slides. Basically, I showed in the mechanism of this strobilurin that strobilurins can reduce the ATP synthesis even in the plants. So when the ATP activity is decreased, it delays the proton pumping to the apoplast, which causes a brief decrease of pH. In other words, because of the decrease in pumping of the positively charged proton, the cytosol becomes acidified. Because of this acidification, the activity of nitrate reductase activity increases. Please remember, nitrate reductase activity requires an acidic pH. This acidic pH is induced inside the cytoplasm because of the lack of protons. Why protons are not available? Because the ATP biosynthetic pathway is partially compromised. That is also the source for protons. So when protons are not available, the cytoplasm becomes acidic, nitrate reductase activity enhances. So this physiological activity is known. But really, does it happen? our studies show that yes, nitrate reductase gene is actually enhanced and the pathway is induced. Then another important gene or pathway that we identified was the brassinosteroid biosynthetic pathway. Brassinosteroids are basically compounds which can prevent infection by a pathogen. In other words, they are very responsible for the defense of the plant. And we found that these brassinosteroids are also enhanced by the pyroclostrobin in tomato. You can see all the green boxes. Now, what are the circles that I have shown? I must mention about them. This particular gene that I have circled here, this gene we have validated. See, this is a pathway that we derived from KEGG analysis. But is it true that this pathway is really running, or is it induced in tomato? How do we confirm? To confirm that this pathway is really running, you must pick up some key genes from this pathway and try to amplify them in the PCR and see whether the gene is actually there or not. That is the actual confirmation that the pathway is running. And that is why I have circled the genes that we have chosen for the confirmation in the future. So we chose the Rubisco, we chose the GA gene, that is the gibberellic acid synthetic gene. We chose the nitrate reductase NR. We also chose the C724B gene that is responsible for brassinosteroid biosynthesis. So having selected all the four, in fact, I told you we identified around 11 metabolic pathways. So we designed 11 sets of primers for the PCR. This PCR is slightly different from normal polymerase chain reaction. This is called quantitative PCR or qPCR. For those who have no idea of what is a qPCR, this is a quantitative PCR in the sense we can precisely quantify what is the quantity of the starting material of our RNA by using this PCR. In normal PCR, that is not possible. Normal PCR is called endpoint analysis. We finish all the amplification in the PCR, then we run the gel. So we get that product doesn't tell us what is the starting material. But qPCR can tell you what is the concentration of the starting material. So we validated all the genes we picked up for each pathway. These are the genes that we have picked up for each pathway. We can see the gene CYP here, we can see the GA2 here, we can see the NR, we can see RBC, that is for Rubisco. Apart from that, we also picked up 7 additional genes. So totally 11 genes were picked up, candidates for validation through qPCR. This is a workflow for the qPCR. We isolated the RNA by using the trisol method that we have standardized in our lab. We synthesized the cDNA by reverse transcriptase kit. So RNA was converted into cDNA. Then we went for quantitative real-time PCR in a Biorad CFX Connect machine by using CyberGreen technology. Then we analyze our data by using delta-delta CQ. That is a standard curve method where we run a standard and we run our sample and we compare the concentration of our sample against the standard. So that is why it is called delta-delta CQ. CQ stands for the threshold cycle value. Okay. Now we analyzed all the 4 genes and you can see the data here. Nitrate reductase, in treated it was increased. CYP, it was increased. Gibberellic acid, it was increased. Rubisco, it was increased. So we predicted in the KEGG analysis that these pathways are getting enhanced by pyroclostrobin. That is true. That is really happening. It is not just we are saying on our own. So that's how we confirmed it. Now this is a summary of the effects of pyroclostrobin on tomato from our study. Pyroclostrobin basically inhibits the electron transport both in the fungal as well as in the plant mitochondria. Because the fungal mitochondria has a very, very low threshold value of pyroclostrobin sensitivity, it dies and therefore the pathogen is killed. But the plant can tolerate this level of pyroclostrobin, though there is a small reduction in ATP production. But overall, because of this, there is a net increase in photosynthesis. There is a low cellular ATP level, which leads to nitrate reductase activity. Nitrate is converted into nitrite. So in the availability of nitrogen and more of carbon, the plant is able to produce better growth and better product. In that the nitrite will also release a signaling molecule called nitric oxide inside the plant. This signaling molecule will induce several signaling pathways, including an antioxidative pathway where it will scavenge harmful antioxidants which are produced inside the plant cell. It will also activate the defense of the plant. On the contrary, this nitric oxide will block any kind of stress that is induced by ethylene, because it will block the ethylene production. Due to high temperature or very low temperature, or due to non-availability of water, if the plant undergoes stress, that stress response will be controlled by nitric oxide and the plant will be able to survive better. And this is all what our pyroclostrobin is doing in tomato. This is how I have summarized. The next slide will conclude my talk. What we found from our study is that enhanced water uptake, because we saw that the xylem vessels are much better in the treated plant, and increased nitrogen assimilation because of reduced ATP level and increased nitrate reductase activity. Increase in nutrient because of better photosynthesis. Enhanced defense because of the production of brassinosteroids and other compounds. Increased carbon fixation and reduced senescence due to blocking the ethylene synthesis. All this lead to better growth, yield, and resistance to pathogens. Now, that is why I told you in the beginning that this is a smart compound. This compound has multifunctions. It is not only a fungicide, can do many things inside the plant. In fact, further studies are required to analyze the effect of this compound in detail on the plants before we come up with some kind of recommendation. Not only that, it is also essential to combine other pyroclostrobins and other molecules with this pyroclostrobin to see what is their effect on the plant, so that we get a compound which is ultimately the best option for enhancing productivity in horticultural crops. With that, I would like to stop my presentation, and if there are any questions, I'll be happy to answer.
The topic is now open for discussion.
If there are no questions, then it can mean only two things. That everything has been understood well by people, or people are not understood.
Sir, thank you, sir. Sir, it's better you can post your mail ID in the chat box so that the participants, if they have any questions, they can contact you personally, sir.
Yeah, certainly. I'll do
okay, thank you. Thomas, madam, Betty Thomas, madam, we can move on to the next.
Sir. Sure, sir. Thank you so much, sir, for your elaborative talk on the compound strobilurin. Your description on plant types such as smart plants and multifunctional compounds are really awesome. Your rationalized views on agriculture and farmer-friendly approaches are undoubtedly useful to mankind. Thank you once again, sir.
Thank you so much. Thanks a lot.
Now we are moving on to the second technical session of this webinar. I have a very pleasant duty of introducing our second guest speaker, Dr. R. Karvenbu Sir, Professor, Department of Chemistry, NIT, Tiruchirappalli, a man of simplicity. Sir is one among the prestigious alumni of Bharatiyar University and has received several awards and honors such as UK India Staff Exchange Fellowship, Young Scientist Fast Track from DST, etc. He has visited several countries and published more than 150 articles in various national and international journals. He has reported more than 20 crystallographic structures. He is a reviewer of several reputed journals of scientific importance. Sir has completed 9 research projects funded by CSIR, TEQIP, DST, BRNS, DAE, DRDO, CARS. Sir has a patent for his work on manufacturing method of metal nanoparticle catalyst. His research interest areas and topics are coordination chemistry, DNA and protein binding and biological studies of metal complexes, organometallic compounds, nano, green, metal enzyme compo, and coordination catalysis, and heterogenation of homogeneous catalyst. And now, I humbly request Dr. R. Karvembu sir to deliver his lecture on the topic nanomaterials in catalysis. Please, sir.
A very good morning to all of you. Thank you, madam, for your kind introduction. Madam, yeah, can you hear me clearly?
Yes, sir. You're much audible, sir.
Can you see my slides?
Sir, yes, sir.
Thank you very much.
Thank you, sir.
At the outset, I thank the organizers, particularly Dr. Palsami and Dr. Sangeeta, for inviting me to deliver a lecture. You must be knowing the application of nanomaterials in various fields. You name a field of science, engineering, or medicine, you can find applications for nanomaterials. It is because of the superior properties of the nanomaterials, they find application in various fields of science, engineering, and medicine. As a chemist, we have utilized the superior properties of nanomaterials in the field of catalysis. The phrase catalysis was coined by the scientist Berzelius in 1835, who was the first to note that certain chemicals can speed up the chemical reaction. So it is very clear that this is not a new field. No, this is a very, very old field of science. The first Nobel Prize to the field of catalysis was awarded to Ostwald in the year 1909, who investigated chemical equilibrium and velocity of some chemical reactions. Later in 1918, Haber received Nobel Prize for his process for the synthesis of ammonia. This is a very important process because this has made revolution in industries, or this has made revolution in agriculture. Only after the development of this process, ammonia could be produced in bulk quantities. Then it was used for the manufacture of urea, and then the manufacture of urea in large quantities. That has made revolution in agriculture. In between 1918 and 2000, there were many Nobel Prizes to the field of catalysis. I skipped all those. Recently, in 2001, Knowles, Naori, and Shortplus, they shared Nobel Prize for their contribution towards catalytic asymmetric synthesis. Knowles and Naori, they have developed efficient catalytic asymmetric reduction processes, and Shortplus has developed, efficient asymmetric oxidation process. Then, in 2005, Krebs, Schrock, and Shavin, they shared Nobel Prize for the development of carbene complexes for efficient metathesis reactions. So it shows that the field of catalysis is very, very important. Many Nobel Prizes have been awarded to the field of catalysis. I have listed only a few of them here. You must be knowing some other Nobel Prizes for this field. And you go to any chemical industry, it may be a pharmaceutical industry or fine chemical industry or cosmetics. Now you can find a separate R&D division for developing catalysts for their own processes. The reason is that developing a new catalyst is related to the cost of the particular chemical or cost of the particular product. For example, the particular chemical is produced by carrying out the reaction for 12 hours. Suppose a new catalyst is developed, so that can speed up the particular reaction. So by using the catalyst, if you can produce the same chemical in 6 hours, then you are doubling the productivity of the chemical by using the same resources. So the cost of the particular chemical will come down, right? So the field of catalysis is very, very important. that is related to cost of any chemical. Yeah. So how catalysts work and why in presence of catalyst, no, the velocity of some reactions they got enhanced. No, the catalysts are workhorses of chemical transformations in industry. About 90% of the chemical products that we use in day-to-day life are made by means of catalytic processes, right? So most of the catalytic processes, no, they occur under industrially feasible conditions of pressure and temperature. In presence of the catalyst, the reaction follows a different pathway for which the energy required is less compared to the pathway, no, compared to the energy of the uncatalyzed pathway. For example, now you just look at this diagram, you can see that no, there is a boy and a girl, and the destination for both of them is this point, this flag, right? The boy no, is trying to reach the destination by climbing over this mountain. Whereas this girl, no, she is very clever because she is carrying a catalyst. No, she wants to reach the destination through a different pathway. She follows no, this tunnel to reach this destination. So it is very clear that this girl no, will reach the destination quickly, or she will reach the destination before the boy reaches, because she follows a different pathway, and for that pathway the energy required is very less, right? Catalyst works in the same way. In presence of the catalyst, the reaction proceeds in a different pathway for which the required energy is less. So because of that, the reaction completes in a shorter time. And in general, catalysis is classified into two types, homogeneous and heterogeneous. Homogeneous is one where the substrates and catalyst are in the same phase, maybe in the solid state or liquid state or in the gaseous phase, but all of them are in the same phase. In the case of heterogeneous catalysis, no, the catalyst and substrates, they are in a different phase. For example, you have a solvent, you are adding substrate A and substrate B, both of them soluble in that particular solvent, so they are in the liquid phase. And now you are adding catalyst, you assume that catalyst is insoluble in that solvent, so catalyst remains in the solid state. Whereas the substrates, I mean reactants, they are in the solution phase. Now the catalyst and substrate are in different phase. This is heterogeneous catalysis. If the catalyst is also soluble in that solvent, now all of them are in the solution phase, so that is homogeneous catalysis. Now some reactions are catalyzed by homogeneous catalyst and some reactions are catalyzed by heterogeneous catalyst. We cannot say always this is better or this is bad. Each of these has its own advantages and disadvantages. I would like to compare these two systems. What are the advantages of homogeneous catalysis? Homogeneous catalysts are generally they're active and selective. What do you mean by active? It means they reduce the time of the reaction considerably. The catalyst is highly active, or the energy required for a particular reaction, it comes down drastically. So it is highly active. What do you mean by selective? In presence of the catalyst, or catalyst not only increases the velocity of the reaction or rate of the reaction, sometimes it increases, not sometimes, most of the times it increases the selectivity of the chemical process. It means, in a chemical reaction, if more than one product is possible, in presence of the catalyst, one product is selectively formed. If that selectively formed product is the desirable product or the chemical that we want, right, then the process is very economic or the process is very good. So the homogeneous catalysts are highly active and selective. So though they are active and selective, right, there is an important disadvantage. That is, since the catalyst and the reactants are in the same phase, and even once the reaction proceeds, product is formed, most of the times products are also in the same phase, right, in the solution phase, for example. Now, at the end of the reaction, separation of the catalyst from the reaction mixture is very difficult, or separation of the product in pure form is very, very difficult. You have to adopt some sophisticated chromatographic techniques. That is possible in laboratory scale, but in industrial scale, no, the adopting chromatographic techniques for purifying the compounds will enhances the cost of the product, and that may be impossible in some processes, during the large scale synthesis. So because of the cumbersome product purification, even though these systems are efficient, these could not be commercialized. Coming to the heterogeneous system, the advantage is that the disadvantage that we have discussed in the homogeneous system, that has become advantage in heterogeneous system. Since the catalyst is in the different phase, you can easily separate the catalyst at the end of the reaction. For example, if the reactants and products are in the solution phase, catalyst is in the solid phase. So once the reaction is completed, just by means of simple filtration or by means of centrifugation, the catalyst can be separated from the chemical process. So this is a very economical process by means of which you can obtain the pure product. You can separate the product in the pure form. That is the main advantage of the heterogeneous catalyst. Then what is the disadvantage here? Right? So this is not active and selective like homogeneous catalyst. Why this is not active? In homogeneous, since the catalyst and the reactants are in the same phase, now the catalyst interacts with the reactants effectively. So now the reaction is very faster. But here the catalyst is in the different phase. The interaction between catalyst and the reactants is not that much. So the process is not efficient as compared to homogeneous system. So now we want a catalytic system that has the advantages of homogeneous and heterogeneous systems. What do you mean by it? I want a catalytic system in which the catalyst should be easily separated at the end of the reaction like heterogeneous system, and the catalyst should be active and selective like homogeneous system. So the advantages of these two systems should be combined, so that will form a new catalytic system. Now, nanocatalysis. No? So what do you mean by nanocatalysis? When nanomaterials are used as a catalyst in a chemical process, then the process is called as nanocatalysis. Nanocatalysis bridges homogeneous and heterogeneous catalysis. How? Right? Nanocatalysts are active and selective like homogeneous catalyst, and most of the inorganic based nanomaterials are insoluble in solvents. So the catalyst can be easily separated at the end of the reaction by means of simple physical process like heterogeneous catalysis. So nanocatalysis combine the advantages of homogeneous and heterogeneous catalysis. This is mainly because of the superior property of the nanomaterials. Because of the very small size and high surface area, the contact between reactants and catalyst increases drastically. This is close to homogeneous catalysis. Why homogeneous catalysts are active? I told you, since homogeneous catalyst and reactants are in the same phase, there is an effective interaction between them. And here, because of the high surface area of the catalyst, there is a very close interaction or close contact between the catalyst and the reactants. So this situation is very close to homogeneous catalysis. So nanocatalysts are active and selective like homogeneous catalysts. And because of the insolubility of most of the nanomaterials, now at the end of the reaction, the catalyst can be easily separated or product can be separated in pure form. So with this background, we wanted to develop some effective nanocatalytic systems for important organic transformations. So initially, we have used well-known copper oxide nanoparticles as catalyst for N-arylation of benzimidazole. You just look at, these are commercially available N-arylated benzimidazole. So I want to say why this chemical process is very important, why N-arylation of benzimidazole is very important, and why we need to develop catalyst for this process. You look at, this is commercially available antiviral compound no, it contains an arylated benzimidazole structure. This is OLED material. This contains an arylated benzimidazole. And this is commercially available drug for hypertension, telmisartan. This is also, this contains N-substituted benzimidazole. I just show you the structure of benzimidazole first, of course, yeah. So this is a benzimidazole, right? So this imidazole is fused with a benzene ring, and this is N-arylated benzimidazole. There's also possibility for C-arylation, but we want to do selectively N-arylation of benzimidazole. And this reaction is very important because some of the commercially available chemicals contain N-arylated benzimidazole moieties. And for this reaction, we have prepared copper oxide as a catalyst by means of a very simple process. Copper acetate was used as a precursor. By using a known method, a copper oxide a nanomaterial was prepared, and that was characterized by means of various techniques. Now, the XRD pattern of the prepared copper oxide matched well with JCPDS data that showed that the formed material is copper oxide, and using Scherer equation, the crystallized size of copper oxide was found to be 8 nanometer. TIM images showed the spherical particles of copper oxide, and these copper oxide nanoparticles are highly dispersed. And surface area was found to be 81 meters square per gram, and this copper oxide is highly stable. It is stable up to 927 degrees centigrade. It means that you can use this as a catalyst even at very high temperature. Of course, we did not use high temperature, but this can be used in a process that is carried out even at 900 degree centigrade. And this is the general representation of N-arylation of benzimidazole. So here different aryl halides were used. We have used aryl fluoride, aryl chloride, aryl bromide, containing different substituents like electron withdrawing group or electron releasing group. The first step in the development of any catalytic process is the optimization of reaction conditions. We have optimized different reaction parameters. Now, we found that 24-hour is the optimum time for the reaction. Even less or more than 24-hour did not yield the good amount of the N-arylated benzimidazole. We have tested reaction with the different bases, and we have found that potassium carbonate was found to be efficient, and 2 millimole of potassium carbonate was required for good conversion of the reaction. Similarly, reaction was carried out in different solvents. Dimethyl acetamide was found to be a good solvent, and the reaction proceeded well at 120 degree centigrade. 10 mole percentage of catalyst was required for good conversion of the reaction. So these are the optimized conditions. By taking one reaction, by changing the parameters, we have optimized the reaction, and under the optimized condition, by using the optimized temperature, base amount, catalyst amount, we have extended the scope of the reaction. We have fixed benzimidazole. We have used different derivatives of aryl halides. As I told you, we have used fluoride, bromide, and chloride with different substituents in different positions. Here, substituent is in the para position. Here, it is in the ortho position. Of course, ortho-substituted aryl halides gave a moderate yield, around 60%. Even the 70% is a very good yield in this reaction. It depends on the reaction. But N-arylation of heterocycles, even if you get 70%, there is a very good yield. So these numbers indicate isolated yield of the product. So different by using the aryl halides with the different substitutions, we could prepare a different benzimidazole derivatives or various benzimidazole derivatives. So if the aryl halide contains electron withdrawing substituent nitro, the reaction proceeded very effectively. 92% of the N-arylated benzimidazole was obtained. Right? So these N-related benzimidazole derivatives were isolated and they were characterized by NMR and mass spectroscopic techniques. And even heterocyclic aryl halides can be used here for coupling with benzimidazole. This is actually carbon-nitrogen coupling reaction, right? So this is very important. If heterocyclic aryl halides, no, this may not be possible in most of the homogeneous systems, because if we use heterocyclic aryl halide, because they contain potential donor atoms, no, these two contain a nitrogen donor atom, this contains sulfur donor atom, so they form easily coordinate bond with the metal that subsequently deactivate the catalyst. But here these donor atoms, they do not form bond with the metal, so a catalyst, the activity of the catalyst remains even if we use heterocyclic aryl halide in the CN coupling reaction. Even you just look at here, 93% of this benzimidazole derivative was obtained when this bromopyrimidine was used as a coupling partner or used as a alkyl halide. So the reaction is, we can use any aryl halide for coupling with benzimidazole to form N-arylated benzimidazole by using copper oxide as a catalyst. So here we wanted to see whether the copper oxide can be used as a catalyst only for the N-arylation of benzimidazole, or it can be used as a catalyst for the N-arylation of any heterocyclic compound containing NH moiety. For that purpose, we have tested the N-arylation of imidazole, carbazole, pyrrole, and indole. All of them contain NH. You see that all, N-arylation happened in all these cases with a very good yield. It shows that copper oxide can be used as a catalyst for N-arylation of any heterocyclic compound that contain NH moiety. So this is a very general catalyst. So any kind of aryl halide can be used. Any kind of heterocyclic compound containing NH moiety can be N-arylated by using copper oxide as a catalyst. So this is a very general catalyst that can be used for N-arylation of any heterocyclic compound. In order to justify the role of nano copper oxide in the reaction, we have also used bulk copper oxide as a catalyst, for which the particle size is 314 nanometer. Actually, for the copper oxide that we have synthesized, the particle size is 8 nanometer. This is the bulk copper oxide that has 314 nanometers. Consequently, you just see that the surface area of, so this bulk copper oxide is 18 nanometer, right? Now, there is a drastic decrease in the surface area of the bulk copper oxide. That is reflected in the isolated yield of the N-arylated benzimidazole. And you see the yield when bulk copper oxide was used as a catalyst was only 52%, whereas the yield of the same compound when nano copper oxide was used as a catalyst was 86%. So it clearly indicates that nano copper oxide, or in general nanomaterials, they exhibit enhanced catalytic efficiency compared to the bulk materials because of its large surface area that interacts with the reactants very closely like homogeneous. So nanoparticles are active like homogeneous catalysts. And the catalyst can be easily separated at the end of the reaction like heterogeneous catalyst, since copper oxide is insoluble in the solvent that we have used, that is dimethylacetamide, right? So the catalyst can be easily separated and it is used in the next reaction. And after the completion of the catalyst, again it is isolated, and again in the third reaction it can be used. Again, it can be used in the fourth run, right? So the same catalyst can be recovered and it can be used for many cycles. It means, the process is very cheap. You just prepare once and you can use it for many times without any loss in the efficiency of the process, right? So this is like heterogeneous. So this shows that nanomaterials or nanocatalysis combine the advantages of homogeneous and heterogeneous. They are active like homogeneous, and they can be separated easily and can be reused like heterogeneous catalyst. And we have proposed a mechanism here, and this is only a possible mechanism. Both aryl halide and heterocyclic compound, they got adsorbed on the surface of copper oxide, and then so the aryl and heterocyclic moiety, they interact together, form N-arylated heterocyclic compound. Then after getting a very interesting results, after knowing the importance of nanocatalysis as catalyst in organic transformation, we wanted to utilize the same catalyst, that is copper oxide nanoparticles, in another important reaction. That is Ullmann type coupling reaction between aryl halide and phenol. So that will result in the formation of diaryl ether. So instead of phenol, if thiophenol is used, then the reaction will yield diaryl thioether, right? So this is not exactly Ullmann coupling, but this is like Ullmann coupling reaction. And so by means of the coupling between aryl halide and phenol or thiophenol, diaryl ether or diaryl thioethers can be prepared. And why we need a catalyst for this reaction? Diaryl ether and diaryl thioether, they are very important compounds. I just tell you only two examples. You just look at, this is the structure of thyroxine. So thyroxine is secreted in thyroid gland. This thyroxine contains diaryl ether structure. Similarly, and this is a drug molecule for a lung cancer that contains diaryl thioether structure. So there is a need for the development of a catalyst for the production of diaryl ether and diaryl thioether. So we have utilized the same copper oxide nanoparticles as a catalyst for the coupling between aryl halide and phenol or thiophenol, which by means of which we have produced a different or large number of derivatives of diaryl ether and diaryl thioether. You just look at the diaryl ethers were formed with the yield of 65 to 92%, very good yield. And again, diaryl thioethers were formed with the yield of 70 to 92%. Here, different derivatives of phenol were used, different derivatives of aryl halides were used, and here also different derivatives of aryl halides were used. And similar to the previous system, we have optimized the reaction conditions. Then under the optimized reaction conditions, different aryl halides were coupled with phenol to produce different diaryl ether derivatives. Now all these diaryl ether derivatives were obtained in good yield. All these compounds were isolated. These values are isolated yields. As I told you, these products were characterized by means of NMR and mass spectroscopic techniques, right? And similarly, under the same optimized conditions, aryl halides and the thiophenol were coupled to produce diaryl thioether in good yields. These diaryl thioethers were also isolated at the end of the reaction. So we could prepare about 30 compounds by using copper oxide as catalyst here. Now from the previous system, from the N-relation of benzimidazole, we came to know that copper oxide is very active like homogeneous. We wanted to test the selectivity of copper oxide in this system, in this Ullmann type coupling reaction. Before that, here also we can use heterocyclic aryl halides like 2-bromopyridine or 2-bromopyrimidine can be used to couple with phenol or thiophenol to produce diaryl ether or diaryl thioether with heterocyclic ring. Because you need, preparing compounds with heterocyclic ring is very important because they have a lot of applications in pharmaceutical industries. Yeah. So to demonstrate the selectivity of copper oxide nanoparticles, we have done two different reactions. In the first reaction, we have demonstrated the reduced selectivity of the reaction. And here, the phenol was coupled with aryl halide. Now, this contains two different halides in the same molecule. This has chloride as well as bromide. So HBr can be removed or HCl can be removed, right? If HBr is removed, then you get diaryl ether, or a chloro derivative of diaryl ether. If HCl is removed, you will get bromo derivative of diaryl ether. So what we have obtained is chloro derivative of diaryl ether selectively. No, the selectivity is 100%. In this reaction, only HBr is removed. The bromo derivative of diphenyl ether is obtained selectively. This product is not at all formed during the reaction. So this showed the selectivity of copper oxide catalytic system. So this is not only reactive like homogeneous, this is also selective like homogeneous catalytic systems. To establish the chemoselectivity of the system, equal amount of phenol and thiophenol were taken, along with the aryl halide. Now this aryl halide now has equal chance to react with phenol and thiophenol, right? So what we have obtained? Now we have obtained this diaryl thiether selectively compared to diaryl ether. So this diaryl thiether is formed with 92% yield, whereas the diaryl ether is formed only with 8% yield. It means the reaction is chemoselective. It means the aryl halide, even in presence of phenol, it prefers to react with thiophenol. So that leads to selectivity of the process, right? So nanoparticles are active as well as selective in important organic transformations. So at the end of the reaction, the catalyst was separated, and that could be used for many cycles without loss of efficiency. Similar mechanism has been proposed for this Ullmann type coupling reaction also. After knowing the catalytic activity of copper oxide nanoparticles in two different organic transformations, we move to another copper catalyst, that is copper on boehmite. Boehmite is nothing but aluminium oxyhydroxide. This is copper on aluminium oxyhydroxide or copper on boehmite. So the precursor for copper is copper 2 chloride, and the precursor for aluminium oxyhydroxide is aluminium tri-secondary butoxide. Pluronic P123, that's a tri-block copolymer, was used as a surfactant, right? And this copper on boehmite was used as a catalyst in the case of oxidation of alcohols to corresponding carbonyl compounds. This is a very simple organic transformation, but very important one. Now, if you look at the total synthesis of any compound, at least 2 or 3 steps involve selective oxidation of alcohols, right? So there is a need for the development of efficient catalytic system for selective oxidation of alcohols. We have used this copper on boehmite as a catalyst in oxidation of alcohol, and we have found that this copper on boehmite could effectively catalyze oxidation of alcohols at room temperature. That's very important. Even in the previous case, copper oxide catalyzed Ullmann type coupling to produce ether or thiether. Now that reaction was carried out at room temperature. Incidentally, that was the first room temperature Ullmann type coupling reaction that was possible with the help of copper oxide. That shows the efficiency or activity of nanoparticles as catalyst. So here the 10 images of copper on boehmite showed the background. The white surface is the boehmite matrix over which you can see the copper clusters, and the amount of copper was found to be 3.21 weight percentage, and the XPS spectra showed that copper, the prepared material not only contains only metallic copper, it also contains traces of copper 1 oxide and copper 2 oxide. Now the stability of copper is not too much. You know very well that when copper is exposed to air, it gets oxidized to copper oxide, right? So this catalyst should be stored in a vial containing inert atmosphere. So we have prepared the catalyst and we have used the catalyst immediately, right? Of course, this is a kind of disadvantage, but considering the efficiency of this material as a catalyst, I don't think this is a big disadvantage. The bed surface area of copper on boehmite was found to be 160 meter square per gram. And incidentally, water was found to be a very efficient solvent for this reaction. You compare, the reaction did not proceed well in organic solvents, but in water, I mean, it yielded very good yield of carbonyl compounds. So it has become advantage for us, right? Room temperature, water is the solvent. So this is definitely a green oxidation of alcohols. No, that was possible because of the nanomaterials as catalyst, no, in this reaction. And we have optimized the reaction conditions. Here, different oxidants were tried. Pyridic acid was found to be the best oxidant for the oxidation of alcohol to carbonyl compounds in presence of this copper on boehmite catalyst. So under optimized conditions, different alcohols were converted to carbonyl compounds. Secondary alcohols were converted into ketones. Primary alcohols were converted into alcohols. You just look at, these values are yields, I mean GCMS yield, whereas the values in the parentheses indicate isolated yield of the same compound. Several benzylic alcohols were converted into carbonyl compounds. Even aliphatic alcohols were converted. Alicyclic alcohols were converted. Even the highly strained bicyclic alcohols were converted into corresponding carbonyl compounds. But in this case, because of the high strain or steric, the conversion is very, very less, but highly selective. Now you just look at the N11, that is very important. Because we can understand selectivity of the catalytic system here. In N11, I mean, there is an oxidation of alcohol to carbonyl compound, but there is no oxidation of C double bond C. Sometimes that will also oxidize to epoxide, right? But here only alcoholic group is oxidized to carbonyl, not olefinic to epoxide. So the selectivity is 100%. Even though the conversion is very less, 34, right? Because it is very difficult to oxidize the aliphatic alcohols, but the selectivity is very good. Only alcoholic group is selectively oxidized. So the copper on boehmite can be used as a selective catalyst under very mild conditions, room temperature, water as a solvent, for the oxidation of alcohols to corresponding carbonyl compounds. Again, to demonstrate chemoselectivity of this particular catalyst, now we have taken equal amount of primary and secondary alcohols, I mean benzyl alcohol and 1-phenyl ethanol. So both can be oxidized to equal extent by means of this process, but it did not happen so. If you look at the products, the benzyl alcohol was preferentially oxidized to benzaldehyde. So we have obtained 83% of benzaldehyde. Whereas the oxidation of 1-phenyl ethanol to astrophenone, now the yield of astrophenone is only 13%. It means the benzaldehyde is selectively formed from benzyl alcohol during the oxidation, right? So it means that even in the presence of secondary alcohol, primary alcohol is preferentially oxidized. So in a particular substance, in a one molecule, if you have both primary and secondary alcoholic groups, by means of this system, you can oxidize only primary alcohol to aldehyde, right? You need not protect the other alcoholic group. You need not protect and deprotect, so that will reduce the number of steps involved in the particular reaction. So after the end of the reaction, the catalyst can be easily separated and used in further processes without a much decrease in the yield. And this heterogeneity test, that is very important here. In the first case, copper oxide, we have used only copper oxide. We did not use any solid matrix to support copper oxide nanoparticles. But here, we have used boehmite as a solid support. On the surface of the boehmite, copper nanoparticles were impregnated. Why? Because if you don't have solid matrix, under the reaction conditions, copper nanoparticles will get agglomerate, so the efficiency of nanoparticles will come down drastically. So to avoid the agglomeration, the copper nanoparticles were impregnated over a boehmite matrix. Sometimes what happens during the reaction, the copper nanoparticles leach from the solid matrix, they go into the solution and catalyze the reaction homogeneously to see whether this is really heterogeneous or some homogeneous catalysts are also there. We have done this heterogeneity test. The reaction was carried out for 1 hour. At 1 hour, the conversion of the reaction was 60%. After 1 hour, the catalyst was removed from the reaction vessel, but the reaction was proceeded without the catalyst, right? So it means without the catalyst, no, the yield did not improve. It means that there is no copper in the solution. Since you have isolated the catalyst, the reaction did not proceed further. It means there's no copper in the solution. That indicates there is no leaching of copper from the solid matrix. Now, this is the proposed mechanism. This is the catalyst copper on the matrix of boehmite. This on reaction with pyroidic acid gives copper oxo species, which is actually responsible for the conversion of alcohol to carbonyl compounds. We have detected the presence of copper oxo species in the reaction by means of U-visible and FTIR spectroscopic techniques. Then, after establishing the use of copper on boehmite as catalyst in the oxidation of alcohols to corresponding carbonyl compounds, we have used the same reaction in the production of beta-ketoenamines from beta-diketones. Now, initially, copper oxide was used was used as a catalyst in two different reactions, in the NR of heterocycles and in the Ullmann type coupling reaction of phenol with aryl halide. Similarly, here, this copper on boehmite, we tried in two different reactions. Why? We use the same catalyst in two different reactions just to show the versatility of the catalyst. You have one catalyst, if that could catalyze only one particular reaction, then you should have as many as catalysts for carrying out many reactions. But if you have one catalyst that could catalyze 4 or 5 reactions, that would be really economical. Industries would prefer it. So that is a versatile catalyst. So here, copper oxide catalyzes more than one reaction. This copper on boehmite also catalyzes more than one reaction. This copper on boehmite was used as a catalyst in the production of beta-ketoenamine from beta-diketone and different amines, right? Initially, the reaction conditions were optimized. Now, the amount of copper in copper on boehmite is 3.28%. We have prepared the copper on boehmite with different weight percentages of copper. This one is without copper, only boehmite. The boehmite did not catalyze the reaction. But if you have copper, it catalyzes. But if you have 3.28% of copper, that is the optimum amount. Even if you have more copper, the efficiency of the catalyst comes down. So we have optimized the amount of copper on the boehmite surface. So that is 3.28%. We have optimized the other reaction parameters like solvent, temperature, base, amount of catalyst, and under optimized conditions, different beta-diketones were used. Also, different amines were used. Aromatic amines, alicyclic amines, even aliphatic amines were used. It's very difficult to to carry the reaction with the aliphatic amines. But you see, this is very efficient catalyst. Within 5 minutes, the 98% of the product is formed. Highly efficient catalyst for the production of beta-ketoenamines from beta-diketones. So different beta-diketones and different amines were used to prepare numerous derivatives of beta-ketoenamines by using this catalytic system. Again, we have tested the heterogeneity test that showed that there is no leaching of copper from the solid matrix. So the reaction is purely heterogeneous in nature. The catalyst can be reused for at least 3 times. There is a slight reduction in the yield of the product at the 4th run, but you can improve the yield by heating the catalyst, so that the reduction in the yield may be due to absorption of some reactant or solvent molecules on the surface of the catalyst, and remove it by heating the catalyst, and then you can again reuse it without the reduction in the yield of the product. And we have also investigated the mechanism of this reaction, that is the formation of beta-ketoenamine from beta-diketone and amine, by means of DFT studies. Now, this is the energy profile of the reaction without the catalyst. This is uncatalyzed reaction. You see, the energy of this transition state, that is transition state 1, is 49.1 kilocalorie per mole. In presence of the catalyst, you see you have the copper cluster. the energy of the transition state has come down significantly. It's only 33 kilocalorie per mole. So as I said initially, in presence of the catalyst, the energy of the transition state comes down. So the reaction is faster, right? And here, there are two possibilities. The copper cluster can interact the ketone oxygen, or it can interact this enolic oxygen. No, it means that, I just tell you one thing here, you have beta diketone. It gets enolized in solution state. So if you have a C double bond O, you also have OH. So the catalyst can interact with ketone oxygen or enolic oxygen. There are two possibilities. So here, the copper cluster interacts with ketone oxygen. Here it interacts with enol oxygen. The energy is less only when the catalyst interacts with ketone oxygen. So it is preferable, right? So the catalyst copper cluster interacts with ketone oxygen preferentially. And I mean, all the transition states were optimized. This is the mechanism. And the beta diketone gets enolized, and amine is added to this double, and the removal of water molecule that finally leads to the formation of beta ketone enamines. Now we have shown that catalyzes two important organic transformations. Then I have shown that catalyzed two important organic transformations. Then later another catalyst, copper to oxide on aluminosilicate, right? So instead of copper, here we have used copper to oxide solid support. I told you, while we have used copper, the stability of copper is question. Avoid that, we have taken copper oxide itself and aluminosilicate, right? And this copper oxide on aluminosilicate is used as a catalyst in a Heck coupling reaction to produce various derivatives of stilbene, and the copper oxide on aluminosilicate will be used as a catalyst for alpha arylation of beta diketones, right? And similarly, the reaction conditions were optimized. We have also extended the scope to various substrates to produce different derivatives of these compounds. And then, after knowing the importance of copper nanomaterials in catalysis, we moved on to ruthenium-based materials. I just take 5 minutes to complete my lecture. Because naturally, as an organometallic chemist, we have interest in ruthenium. So we wanted to prepare some ruthenium nanoparticles so that it can be effectively used as a catalyst. Though it is costlier, the ruthenium is a well-known transition metal catalyst. I mean, the efficiency is very good compared to other non-precious metal catalyst. Here we have prepared ruthenium oxide nanorods on the surface of graphene nanoplatelets. Here, ruthenium triacetyl acetonate was used as a precursor for the formation of ruthenium oxide nanorods. You just look at a very interesting morphology was obtained, and you see it is very small nanorods, so up to 5 nanometer, right? So these ruthenium nanorods or ruthenium oxide nanorods on the surface of graphene nanoplatelets were characterized by means of various techniques, and this was used as a catalyst for transfer hydrogenation reaction. This is like MPV reduction or open air oxidation. Many carbonyl compounds were reduced to alcohols here. Just is opposite to oxidation, right? Here, there is a transfer of hydrogen from alcohol to this carbonyl compound. This carbonyl compound becomes alcohol, and this alcohol becomes carbonyl compound. So this is actually a redox reaction. Both oxidation and reduction happen together. But I mean, of course, the reverse reaction is also possible if we take the alcohol in stoichiometric amount. But we take alcohol in excess, so reverse reaction is avoided. We have kept this 2-propanol as a hydrogen donor in all the reactions, but we have changed this ketone and we have produced many alcohols, even the benzylic alcohols, aliphatic alcohols, alicyclic alcohols can be produced in good yields. And again, there was no leaching of ruthenium from the surface of graphene nanoplatelets. And similarly here, we have prepared, so that is a ruthenium oxide nanorod. Here, ruthenium nanoparticles on the surface of graphene nanosheets. You just look at the size of ruthenium nanoparticles, it is 0.5 to 3 nanometer. No? So because of the very small size of the ruthenium nanoparticles, they are very effective catalyst for the oxidation of alcohols to carbonyl compounds. Why I say it's a highly effective? When we have used the copper on bohemite, we have used pyridic acid as oxidant. But you just look at here, no oxidant is employed. It means that it uses air as oxidant for the oxidation of alcohol to corresponding carbonyl compounds. So this is aerial oxidation of alcohols to carbonyl compounds by means of ruthenium nanoparticles impregnated on the surface of graphene nanosheets. Again, a similar chemoselectivity was observed. The primary alcohols can be selectively oxidized even in the presence of secondary alcohol. And this chemoselectivity has been established with aromatic system as well as with aliphatic system. We have observed the similar chemoselectivity for this ruthenium-based catalytic system for oxidation of alcohols to corresponding carbonyl compounds. Now, once the reaction is over, the ruthenium nanoparticles were recovered from the reaction, and by heating it, again the ruthenium nanoparticles were converted into ruthenium nanorods, and the ruthenium nanorods were used as a catalyst to transfer hydrogenation reactions. That's why I said this is easily tunable ruthenium. You do the reaction. After the end of the reaction, maybe if you want, you can use it as such, or otherwise after the reaction you can convert it into ruthenium nanorod just by heating, and then the ruthenium nanorod can be used as a catalyst in transfer hydrogenation reaction. Very versatile nanocatalyst. I would like to conclude my lecture that nanoparticles, particularly copper-based ones, are cheap, they're air stable, efficient, reusable, and they're heterogeneous system, so the catalyst can be easily separated. The procedure is simple because in a homogeneous system, you need to prepare ligand, you need to prepare organometallic compound. Most of the times they require very sophisticated conditions. So here it is a very simple preparation, ligand free, and they are very efficient to offer products in high yield, operationally simple. I thank DST Nano Mission for financial support, and then I thank Professor Iksu Kim Shinsu University, particularly for collaborating while we have developed ruthenium-based nanosystems. Dr. Ganesh Babu, as my PhD student, who has developed all the copper-based systems that I have shown here. Dr. Kobiraman is actually the PhD student of Professor Iksu Kim. He has developed ruthenium-based catalytic systems. I thank my institute, NIT, for providing all the facilities to carry out my research. Again, I thank all the participants for hearing my lecture patiently. And once again, I thank organizers for giving me this opportunity. Thank you.
Yes, sir. The topic is now open for discussion. Okay, fine. Thank you so much, Dr. Karvimbu, sir, for your determined talk on the topic nanomaterials in catalysis. You enlightened us about the history of catalyst and the two processes of catalysis, and also about the role of nanomaterials in catalysis and its preparation. Really, you have made justice in your part in educating us in the given topic. Thank you, sir.
Thank you, madam.
Moving on to the third technical session. I am delighted to offer the most hospitable, cheerful welcome I can to welcome and invite Dr. N. Ayadure, sir, Principal Scientist, Associate Professor in Biochemistry and Biotechnology, CSIR, Central Leather Research Institute, Chennai. Sir has completed his PhD in Biotechnology in Pondicherry University in 2006. Sir worked as an invited scientist in Biocenter Division, Korea Polar Research Institute, South Korea from 2006 to 2007. In Division of Biomolecular Engineering in Busan National University, South Korea, Sir has completed his postdoctoral research in 2009. From 2009 to 2012, Sir has worked as an assistant foreign professor in the School of Biotechnology, Yangnam University, South Korea. From 2012 to 2013, he worked in the Department of Biochemistry in King Saud University, Riyadh, Saudi Arabia, assistant professor. Sir worked as a senior scientist from 2013 to 17 in the Department of Biotechnology in Central Leather Research Institute, Adayar, and at present, Sir is working as a principal scientist in CSIR, CLRI, Chennai. His areas of specialization are molecular biology, genomics, and protein modification. He has published 65 articles in both national and international journals. He has 3 patents and Sir completed 2 major projects, and now 3 projects are ongoing. Sir has guided 6 PhD students. He has 5 years of teaching experience at various academic bodies. Sir has secured various awards and honors like Young Scientist Award by DST, Uppsala University Award by Uppsala University, Sweden, Riken Invited Grant by Riken Institute, Japan in 2012, Finn World Travel Award offered by the Protein Society held at Boston, USA in 2011, and Wellcome Trust International Award in 2009. Sir is a member of Protein Society, USA, editorial board member in Research Journal of Biotechnology. He is also a member of Korean Biotechnology and Bioengineering Society, Biotech Research India, Associate of Microbiologists in India, and Korean Chemical Society in Republic of Korea. Now, I feel much pleasure to invite you, Dr. Niraikulam Ayadure, sir, to share with us about your views and knowledge on the topic Green Fluorescent Protein and Nobel Prize winner for discovery. Please, sir.
Thank you very much, ma'am.
Thank you, sir.
Audible to all, am I right?
Yes, sir, you're audible, sir.
First of all, it's a great pleasure to be, a part of this ongoing seminar. And I would like to express my sincere thanks to Dr. Sobana for this invitation. We had a talk, and I think of the first one we started with such a very special core about one of the best talk about the translation research by Professor Subramanian. That was very interesting. And the second one was we moved to the chemistry, and we learn about, I think, the importance of catalysis, and especially in the field of nanoengineering. Both are very interesting. They made, both of them made an very important aspects in human life. So now I will take you to some other mode of approach where we can have some story, the biological stories. Especially, I'm going to share some of the inspiring work or inspiring a story of certain inventors who made their path, the end of the day resulted with a novel protein, which lead to one of the very highest prize of the Nobel Prize. So I'll start with, I'm going to share some of the novel discovery, especially I'm going to focus about the importance of green fluorescent protein and how it has been discovered, and later how this protein has been extensively used, and now it is, it's occupied one of the very important reporter or marker for cell biology, especially for several biotechnological applications. You could see this slide, am I right? It's very interesting, and sometimes we can revamp by ourselves. This is a very beautiful seashore in Maldives. When you walk around, the Maldives are very famous for tourists, especially that they have a very good, beautiful seashores where you can spend your time in the evening. And So Maldives is very famous for this kind of tourist spot. But can you see the first slide? There's a very beautiful moon, and you could see the seashore. And when you have the float, it's blue in color. What is that? Oh, we mostly, if you go to our Chennai base, Marina or something, we can't see this one. Normally, we could see the floating. That's all. But it's very unique. You could see that that's very good and blue in color. The second thing also, it's very, they made very intensive colored, and if you look at very little bit, deeper inside, in the seashore, you could see that there are a lot of blue in color. It's something like we usually call the in Tamil terms we use serial bulb, something the LED lights, something like that, right? We could see it in the seashore. What is that really, and why we are getting such a beautiful blue color or some of the illumination in the seashore? That's mainly because of the presence of some of the marine organism that's called jellyfish. I think a lot of people would have seen that one. Maybe as a student, you had a lot of time to visit the seashore at some of the place where the aquarium or any other marine institute. If you go there, you could see that there are jellyfish, which is very unique in structure, as well as it's very unique a morphology of transparent in nature. That is mainly because of the presence of jellyfish in that particular seashore. Okay, when we think about, as I said, it is mainly because of the fluorescent protein producing jellyfish. What is this jellyfish, and what is the relationship between these two protein and the organism? What happens over there? Actually, naturally, the jellyfish it's produced a specific type of protein, which is called a bioluminescent protein. It was first observed and discovered in 1960, and by the people when they go to the seashore, or when they went to the sea, they could see that in the night time there are some marine organism. It's glow even in the water. And why it is? Because the jellyfish it has produced a fluorescent protein. It's one of the escaping mechanism. When higher animal, for example, a bigger fish is going to capture this jellyfish, what it will do is to escape from that fish, escape from the predators. What it will do is immediately it produce a color and change it in nature. So what will happen? This fish may think that, oh, something is wrong, so I don't want to eat that fish. So it will escape mechanism. So evolutionarily, this fish has developed started producing this protein in a stress environment to escape from the predators. Similarly, we could see a lot of similar structure in our life. When we are in the child, we could see that we call that, Minmini Puchi. So many people would have seen that, am I right? In the night, it glows very well. It's very attractive. Why it's specifically glowing in night? That is one of the mechanism of survival. It attracts the mates for reproduction. So that produces some kind of protein which is called luciferin. That luciferin, it's going to bind with the luciferase with the help of enzyme. Then it started glowing. So people has identified, okay, there are some protein which could be used for, some of the protein may be produced by the marine organism, which is very glowing in nature. Okay, what is that relationship? Okay, so what? It produces some protein, and how we can use this particular protein for biotechnological applications. What happened when the scientists started arguing the next stage? I mean to say that earlier day we have a debate that DNA is the coding region for our living system. So the people are started demonstrating the importance of DNA. Later, there is an argument between the scientists. No, DNA is not a major coding organism to play a critical role on any function or metabolism. Only protein is play a critical role. So these are the concepts that evolved on the day. Then people were started, accepting, okay, DNA is genetic material, and later the Watson and Crick has come, and they proposed the structure of the DNA, and people started accepting, okay, DNA is the major compartment for deciding authority of our genetic nature. It happens. Okay, this is a part of things happen, a starting evolution of genomics. In the case of proteomics, what happened? People want to know, okay, some of the proteins are producing, whether these proteins are produced in which organelles. For example, in a eukaryotic system, whether it is produced in the cytoplasm, or it is produced in the periplasm, or it produced any of the organelles, something like a vacuole or a ER, there's an endoplasmic reticulum or something. So they don't know any tool to produce or to identify how the protein could be produced, where it could be produced. So people thought about it. There are two different scenarios going on between these two cases, and people first that day come to know that, okay, there is a protein. This protein is glowing. So what they did is they simply tagged the interesting protein. For example, I believe that some of the PSP protein, that is some of the plant proteins, photosynthetic protein, may be produced in cytoplasm. But there is another arguing that, okay, this particular protein might be produced in the periplasm. So what we did was they conjugated, they tagged this fluorescent molecule with our protein, and they put it in the protein in the cells. Then they see that, okay, if they identify that particular protein is glowing in the cytoplasm. So people at the time they prove that this PSP protein is produced in the cytoplasm. It's not produced in the periplasm. It is something like we also hear during our lifetime, right? You know that we developed a concept when we have a kids. The kids always wear the bangles. I mean the kolusu. Am I right? Or sometimes some of the people will put some kind of bell to the kids to identify them. That's mainly because the mother may be working in somewhere. The kid may go to somewhere places. So to always kids should be the mother should be aware of that the kids is wearing. So she wants to always hear the sound. That is one of the reporters. Similarly, what people has done is protein has been tagged with the reporters, and they put it in the cells, and they identified where it has been expressed, and they propose it. Not only the work has been started in a similar way, and later by understanding the structure and function of this fluorescent protein, they have used it for plenty of experiments. For example, they have used as a thermometer. They also used as a fluorescent dimer, and they also used as a stress detector, and also they used as a neural growth, etc. And nowadays, it is beyond that limit. You could see that they have developed a lot of transgenic animals, like you could see in the field. And there are some the rabbits and a rat, which is expressing the fluorescent protein. So it has a lot of applications. That's why it occupied a specific role in cell and molecular biology. Okay, we will start with discovery. And for this wonderful discovery in 2008, the Nobel Prize has been offered to three people. One is Professor Shimomura, and second one is Professor Martin, and third one is Professor Ashin. So these three people had a lot of breakthrough in this identification and this fluorescent protein. I'll tell you some of the story. Even among out of the three, one more person, that is Douglas, also played very interesting role on that. But of course, he has been recognized. But although he didn't offer the Nobel Prize, he's also one of the person I would like to highlight his discovery in the fluorescent protein. Here, the Simu Roy is one of the inspiring scientists, and I just wanted to share some of his earlier resulted with the discovery. As I said, all the three has had a unique features. Although all don't have each other linked with the research, they have other unique features. Professor he is born out of Japan, actually, and he was a student of Professor Yakumo. And happened when he joined his B medical science, and that's his mentor is he started in MSc and that time what happened, he published a very interesting paper, especially how the firefly and some of the material glowing which play important role on what is the mechanism behind on it. But what happened is, everybody knows in August 1945 the Second World War has started, and especially he was studying in university, the atomic bomb has been attacked by the city. So then he's completely, the Nagaya University was completely destroyed, and he's the person who lost his degree. And after that, what happened? He joined as a MSc in nearby university, but he was working as a part-time student. So, but he was always having an inspiration that he always keep talking with the people that he identified something very unique from the firefly and also from the jellyfish that he want to explore. But at that time, he don't know what is the real mechanism behind on this. So he wrote an email to Professor Johnson in Brigham University, where Professor Johnson was highly attracted and about this project, and he invited him to join as a graduate student in his lab. That's happened in 1961. So what's happened? You just imagine these days, going to US or getting a PhD position is much better and easy. But in 1961 and 1960s, getting a position 2 years, and especially moving from the country like Japan, is not so easy. So he was attracted and he went and joined with the Professor John's lab. And what happened? He proposed a project, and although he proposed he should have the protein, am I right? So what he usually do is he will go to the harbor lab. That's a harbor laboratory based lab. They will go to the seashore with his family. The whole family will go and they will collect them jellyfish, and they will transport that into the small lab near to the present in the Friday Harbor lab. And in the lab, they will start extracting the protein. And because just imagine, he wants to extract more amount of protein. For example, he collected more than billions of jellyfish, and they extracted, and finally he proved that he successfully solved the structure. That is his achievement. He is the one first solved the structure. And once the structure has been identified, then as a protein engineer, we could easily catch up that we could play or tune the protein as what I want. So this happened in the early 1985, 86. So after that, what happened? In 1988, Professor Simon was giving a talk in a conference, and he was explaining about his invention and discovery and solving the structure of this fluorescent protein. And at that particular seminar was heard by Professor Martin, who is the person who was working in C. elegans, and he's the one suffering with like he wants to know where particular protein is really is produced, whether it has been produced in the cytoplasm or periplasm or something. So he was very happy with the presentation given by the Professor Shimomura, and he taken that achievement, and he is the one, along with Professor Douglas, they pull out a sequence of the GFP, and they identify the genetic sequences and the sequence coded for the green fluorescent protein, and they cloned and they expressed in the lab, and they have been used for as a tagging material to find out the expression of the protein. He's the one first did that experiment. In the Nobel Prize meeting, Professor Martin mentioned it very clearly that he couldn't resist after hearing the talk from the Imimura. He said that I was just quite after the conference, I just left the room, went to the lab. I asked Douglas, one of my students, to start the project immediately. That's the inspiring thing happened from the Professor Imimura. Then later, true story, and after that, Professor Roger is joining with us. He's basically born and brought up from China, and now he joined as a professor in Yomiuri University. He is the one understand the first is like propose a structure, and second thing, Martin has developed the clone, and now Roger has identified that the particular fluorescent protein, the fluorescent nature is coming from the 3 different amino acids. So he manipulated all the protein, and he produced different color in nature. He is basically a Chinese samurai, so he can manipulate anything. So he manipulated and he produced a n number of different variant of proteins from the single GFP. That's the achievement. For that, for this in 2008, the Nobel Prize is put to all 3 persons. So one of the inspiring story I always inspired by Shimomura, the person who suffered, and end of the day he got the Nobel Prize. So we will go into the little bit chemistry of the structure of the green fluorescent protein. In green fluorescent protein, it's a protein is by 28 amino acids. One of the very unique feature of this green fluorescent protein is the chromophore, that is the fluorescent, has been formed only by 3 amino acids that present in 65, 66, and 67 place. That's glycine, tyrosine, and glycine. So what's happening? This glycine and tyrosine, it will go for a cyclization followed by oxidation and form a unique imidazole ring containing a chromophore. And what is it? It has a very unique structure. The GFP has a very unique structure. It contains around 11 beta barrel sheets, and inside the beta barrel sheets, we'll have one cylinders which is buried inside the beta sheets, and this, especially the chromophore also buried within the beta sheets. So we call this as one of the unique beta can structure. What happening here is the unique nature of the green fluorescent protein is, as I said, the amino acid present in the 65, 66, 67 always go for a interaction, and it produces the color. What happening is in the 66 position, instead of tyrosine, if I put a histidine, is change the color into blue. Then if I put the tyrosine, then the color is going to be green. And if I change the color to tryptophan, it changes the color into cyan. So when we introduce different colors, different amino acids, we can get different color. That's a very unique feature of this chromophore. And especially this fluorescent protein, it does not require any kind of chromophore. It does not require any kind of substrate for to glow. For example, most of the fluorescent protein, or even the case of luciferin or something, it always need a substrate. So the substrate is, without the substrate, it will not glow. But in the case of green fluorescent protein, it's an autocatalytic in nature. So it will produce by themselves. So we can use it for several applications. As I said, once it has been developed, it has been used as a transcription visualization, and we can specifically detect the small molecule where it go, and how we could use it for detection like in a small molecule can go to the particular place and where we can glow the protein and we can study that. And also we have used this protein for studying the protein folding and refolding efficiency. In our lab, I started working on this fluorescent protein, and we have developed our own fluorescent protein from the lab, and we successfully recombinantly produced, where my fluorescent protein also have a chromophore at 65% is glycine and tyrosine and glycine. It will go for a cyclization and it will produce a green fluorescent chromophore. We started working on it. We extracted the DNA, and we subclone and we clone, and we transferred into the E. coli system, that is a bacterial system. And whenever we need, we express the protein and we will purify it in our lab. For example, just imagine that in the initial stage, Yamimura collected millions of jellyfish to extract a few grams of fluorescent protein. But now, because of the advantages of recombinant engineering, I could clone them and I can produce the same 1 millimolar or 1 gram of fluorescent protein within a day in the lab itself. That is one of the. This could be one of the important highlights of the application of recombinant engineering. So our lab, we produce this fluorescent protein in our in the lab itself. Somebody may ask that, okay, there are a lot of reports like they have introduced different natural amino acids, they changed it and they changed the fluorescent nature also. What is the unique and importance of doing that one in our lab? So we thought that like people have already replaced existing natural amino acids with one another. For example, if the tyrosine is there, tyrosine was replaced with another amino acids like tryptophan or histidine or whatever it is. All are naturally occurring amino acids. Am I right? Our cells produce, or any living system is going to produce 20 naturally occurring amino acids that could be introduced through the mutations. So we thought, why should I limit with the 20 amino acids? What will happen when I'm going to introduce the 21st amino acids? For example, if I ask some of my friends in the nearby lab, chemical lab, I can ask them, can you produce some of the very interesting amino acid analogs, which is our surrogates of natural one? For example, if I want to introduce an additional hydroxyl 1 or a acid group, which is not possible with the universal system. So we asked, and they've given a lot of tyrosine surrogates. That's a tyrosine mimicking unnatural amino acids that was we successfully introduced, and we want to study them. Okay, what will happen when we introduce this kind of unnatural amino acid into the green fluorescent protein? We have introduced, this is a normal one, and this is what people usually do it in the lab. And we thought that why not I can introduce different unnatural amino acids, and what's going to be happen, how the structure is going to be changed. We started with that, and as I said, naturally, if we could develop a biological tracker, what is the important one? If I could develop a biological tracker that could be used for several applications, like I could develop a protein which could be used for neuronal growth studies. We started with our experiment. One of the approach what we are planning is like in order to introduce this kind of natural amino acid, we should know that how this the central dogma is playing. As we know that we have a 20 amino acids, as I said earlier, we also have a 20 specific tRNA synthetase unloaded with the specific tRNA. So here, once the amino acid has been translated and that has been amino acylated, it will site-specifically selectively go and introduce into the codon. And here the important thing is the ribosome does not care what is the amino acid has been charged. It will always care about what is the whether the codon anticodon is pair is fine. It will go and deliver into the protein. To exploiting that one, we have introduced several unnatural amino acid protein, and we have used for studying the protein structure and function, protein-protein interaction. Definitely, we have developed some of the therapeutic proteins. So for that, what we did was we have developed recombinantly modified E. coli system where it's called auxotroph. This auxotroph will be grow at the particular level of medium, and at the particular stage we completely deplete the methionine, and we will add the directed methionine or phenylalanine target natural amino acids to the medium that could be selectively introduced into the proteins. The first application is like when we introduce this natural amino acids, you could see that the figure as a visibly, it's a green color in nature, and once we introduce the natural amino acid, it tragically changed into the orange in color. That's mainly because of when we introduce natural amino acids that change the critical nature of the chromophore that result in the fluorescence shift. Recently, we have developed a very interesting fluorescent protein, which is mainly coded by tyrosine analog. When we introduce this natural amino acids, the protein chromophore, what happened? The green fluorescent has been shifted to red color, which is not very unique, which is not normal. Naturally, we could convert the fluorescent protein to orange or some of the maybe like 30 or 40 nanometer shift, but making a large stroke shift to 500 to 600 is very, very unique. So we have extensively studied what happened when we introduce this amino tyrosine into the protein, and we learned that when we introduced a single additional amino group containing tyrosine, it resulted with excited state of electrons. These electrons are jumped higher in a way, so it resulted in a large stroke in green fluorescent protein. You could see it's very clearly, this is a red fluorescent protein created in our laboratory. When we introduce, before that it's green in color. When we introduce this amino group, what happened? We have successfully shifted into the red color protein. Later, okay, maybe you could think that, what is the application and how this could be used. The red color could be used, or this kind of protein could be used to study as a biosensor, and we could use it as a biomarker. For example, we are planning to use this kind of protein for brain cell imaging, because brain cell is always naturally thicker in nature, so we need red color protein, which is important for the labeling of thicker cell walls, because red color can penetrate deeper than the green color in nature. So we have used that one, and as I said, we have developed a bioconjugation strategy using this functional group with this clean fluorescent protein, and we have developed a unique structure that was going bind inside very interactively. It's a kind of non-enzymatic interactions. So we could use it for protein immobilization and also for protein labeling approaches. Once we developed that one, we have also used as a sensor. The sensor has been used specifically to detect a certain level of metal ions, because selective identification of metal ion is very, very important. Because in any biological sample, it should be maintained at threshold level. When it exceeds higher the threshold level, what happening is it resulted in pathological conditions. So we have developed a unique sensor, and we identify that the interaction is specifically happening by a metal coordination, not by any structural interactions. That has been proved by studying the structural interactions, as well as certain the fluorescent regaining analysis also strongly suggests that this kind of protein modification could be used for metal sensing applications. These are the ones we developed. We have also developed that one in a real sensor. We have developed protein array. That array was used currently it is under evaluation. Once it could be evaluated, and that could be commercialized for copper detection analysis. Summary, in the end of the day, what I'm trying to say here is like we started with the protein. That is, the protein is like very inspiring protein. We identify the person who is inspiring the protein engineers, and we learn the discovery, and later we have applied our knowledge and how this protein could be selectively modified with unique features. When we introduce this kind of unique chemical structure, what happening here is it resulted with new functional moieties. That functional group could be used for several applications. We proved them. It could be used as to study the protein-protein interactions, and it could be used as adhesive properties. That also could be used for metal sensing applications as well as for immobilization. Thank you so much and for your repeation. And if you have any questions, I'm happy to answer. Hello.
Topic is open for discussion. Okay, fine, sir. Thank you so much, Dr. Ayadure, sir, for your elaborative talk on the topic green fluorescent protein. You described very well about the fishes, various life forms, and the protein, the so-called luciferin, a brief history about the previously worked scientists. It's a very perfect example for rDNA technology. Thank you so much, sir.
Thank you, ma'am.
The final technical section on this webinar. Extend your arms in welcome to the future. The best is yet to come. It's my gratification to introduce and invite Dr. Ramasubramanian, sir, Chief Administrative Officer, IIM, Kashipur, Uttarakhand. For about 33 years, he has served in Indian Navy in different ranks up to the rank of captain and as Deputy General Manager, Head, Deputy Head, Area Commander, Control of Examinations, Professor, Registrar, Chief Administrative Officer, HOD, and Vice Principal. Sir is a multidisciplinary specialist by academic qualifications and also by experience in engineering, social sciences, management, and administration. He is a gold medalist in instrumentation. Sir is a postgraduate in marine electrical electronics and communication engineering and holds MTech in avionics. He is a doctorate in electrical engineering. Sir has done MBA in HR management and MSc in applied psychology and holds PhD in psychology. He is a counseling psychologist and a specialist in aircraft and helicopter servicing, maintenance, overhaul, repair, and modernization. For various diplomatic missions, purchase and overhaul of aircrafts, Sir has been deputed to various countries like England and Russia. Sir has been trained at Russian Aerospace Organization on advanced avionics and by Westland Helicopters of England on tactical mission systems. He is a fellow member of Institution of Engineers in the aerospace engineering domain. In Mr. PhD Tech competitions, he was a runner-up. He was a best outgoing student of MIT and a best student of Tamil Nadu, which is awarded by TTK Foundation. He was a General Secretary of NSS in MIT and founder member and chairman of Personality Development Association at Anna University. Sir has been awarded War Medal for Operation Parakram and Kargil War Medal for Operation Vijay by Government of India. Sir also received special commendation from the Commander-in-Chief for meritorious service. For the past 10 years, he has been the visiting professor in Department of Electronics and Communication Engineering in PSG College of Technology, Coimbatore. Sir has written various short stories in Ananda Vigadan, Kumudam, and Bhakya with standard literature. He has published number of articles in various international reputed journals. 1,750 articles are there in his blog, www.funnyramsblogspot.com, for the benefit of all age groups. And now, really from the depth of my heart, I would like to tell you, sir, I'm much privileged to be here to introduce a legendary captain to the gathering, and I would request you and invite you to enlighten us on the topic technology, innovations in marine warfare. Please, sir.
Thank you, ma'am. Thank you so much. Audible?
Yes, sir, you're audible, sir.
Thank you, ma'am. So nice of you. Have a good day. And I'm thankful to Dr. Dhanam, who has given me this opportunity to be here, the head of the Department of Physics of Kongunadu Arts and Science College. Now here we go with the presentation. Can you see my presentation, ma'am?
Yes, sir. Your presentation is visible, sir.
Thank you. Right. Okay. So the topic of this conference is exploration of knowledge on science and technological innovations for nation building. Yes, nation building. That is a key word that I can look at. And of course, we have the science and technology developing in this country for quite some time. So let us focus on this nation building, right? So my talk is related to technology innovations in marine warfare, right? Okay, now having served in the Navy for 35 years and sailed on board ships, submarines, and used all the technologies, and have been the head of organizations which undertake the overall of aircraft and helicopters, repaired a lot of systems, maintained overall, life extended, been to abroad many number of times, and visited all the aerospace organizations in Russia. And so with that background, I thought I'll give some talk. Now the time left is very, very less for me. Right. Anyway, I'll just do some justice. Now everything is science. Being a science student, I did my BSc Applied Sciences from the Faculty of Engineering from PSG College of Technology. Therefore, I'm a science person. Right. I'm convinced that if a country has to be a superpower, it has to be strong in science, with the best of the scientific laboratories, innovations in science and whatnot, right? And of course, I am in close association with Indian Institute of Science and MIT and other places. Though I am now the administrator in the Indian Institute of Management, I'm basically a science and engineer and a technologist, right? Okay, life is a journey of learning. Yes, it's a continuous journey of learning. There is, it is not like you do some studies and then work and earn and stop thereafter. No, it is a journey of learning. We should lose ourselves in what we do. Yes, that is very, very important. I presume that a lot of students are here and scholars are here, and I emphasize that we should lose ourselves in what we do. Otherwise, the outcome of the product or service will not be the best, right? Research is life. It's not an activity or a profession. When I say research, it is not just doing a PhD and finishing it off and say bye-bye. Let me now look at my professor promotion or head of the department promotion or the registrar or vice chancellor promotion. No, right? Research is a lifetime activity, and it is not an active. It's a lifetime activity which continues. It's not a single activity or a profession. Importance of materials and processes. Yes, most of the researchers, when we see, they come to a halt. We come to a into the stumbling block due to non-availability of materials or processes. Okay, this is what I observed, because when I finished my BSc Applied Sciences, my aim was to do MSc Material Science and do MTech at IIT Kharagpur. MTech in, right? That was the path. But subsequently, what happened? Indian Institute of Science and MIT they gave opportunity for BTech after 3 years of BSc through national level merit exam. So I got through at MIT. Therefore, I went as an engineer. I became an engineer. But then my aim was to become a material scientist, right? So that is the reason I keep studying more about material science and things like that, right? Need for researchers in fundamental sciences. It's very important. Otherwise, a country cannot boast itself, a superpower. That's why I'm very much convinced about it, right? Think beyond the boundaries of laws. Yes, it's very important. Now, when we have the problems created with a particular thought or a law or a equation or a philosophy or a theory or whatever, we cannot find solutions within the boundaries of it. So that's why wherever I go, whether it's an engineering college or NIT Trichy or NIT Trichy, yeah, I keep visiting. Yeah, one professor I saw, he gave a lecture now, right? And I am here. I keep telling the students that learn all the theories, learn all the equations, learn all the laws, etc. But then think about disproving them. Okay. At the end, you will have either of two. One is that either you would have proved that it is wrong and you would have become a Nobel laureate or the best guy on the earth, or you would have learned much more than the Einstein or Curie or whoever has proposed the theory or equation, right? So therefore, I keep telling all the students and research scholars that think beyond the boundaries of laws that you have learned. All these things you have learned, not to make strong structures within which you remain, and then this is how it is, this is how it is, this is how, then you cannot make any breakthrough in research or come out with the solutions for the problems that we have. Why am I emphasizing this is that the problems that I'm going to tell you now, till now, the world over, we are not able to solve these problems. Okay, and it is very important to solve these problems. Otherwise, we can't call it as ourselves as military power, superpower, right? If India has to become a military superpower, then we have to have it. So therefore, think beyond the boundaries of laws. Very, very important. Okay. Every outcome of research should not be converted into a product or service. Sadly, what we have done is that we have converted all the products and service, all the outcomes of researchers which are health compromising into products and services. That's why we have all kinds of pollution, diseases and disorders and whatnot we have, right? So therefore, many researchers will continue for development of technology, science, but every research outcome need not be converted into a product or service, that we must remember, right? Economic researchers on recycling of domestic and industrial waste, yeah, these things are very much important today. Eco-friendly fuel power source for automobiles and industries, yes. Eco-friendly dwelling units for human beings is very important as the population is increasing, exploding exponentially. Affordability of research outcomes are poor, it's very, very important. Unless the research outcomes, they reach the poor, the villages, as it was told by many speakers, Dr. Vasuki or the Vice Chancellor, Warrior University or subsequent speakers, it should reach the poor, otherwise there is no use of that research, right? Now, also what happens is, the technology improvement, science developments, etc. should change the society. The people have to change, the parents have to change and motivate the students for lifelong research and studies, rather than telling them, okay, padichachi, okay, now go for a job, get married, produce children, and involved in home or domestic life, right? So that mindset should come in the mind of the parents for motivating the students to go abroad, to go to the best of the places and study, right? Because being a counselor, I've got about 2,000, 3,000 students of CIT, PhD Tech, NIT, Shastra, and other IIMs and IITs under my, wings. When I counsel, this is what apparently comes out, that either the parents or husbands are not allowing the children to explore the intelligence that they have. Children are having too much of intelligence, they can do much more, but then the parents and the husbands are not supporting them. Okay, right, next. So, military superpower, yes, India has to become. Why? Importance of being military superpower, yes, why not? I'll tell you the reason. We have got so many superpowers. If I tell somebody to name the superpowers in the world, Russia, America, Germany, Japan, England, everybody will name, right? And if I tell somebody to name the oceans, everybody will say Atlantic Ocean, Arctic Ocean, Pacific Ocean, all the oceans they will name, but there is one ocean known as Indian Ocean, right? There is no ocean named after any superpower. Okay, but there is only one ocean named after one country, small country in the world, that is Indian Ocean. We have not named it. The world community has named it, not yesterday. It is thousands of years back, when the map was formed, or when the history of the world, world was world started, right? So therefore, India plays a very key role in the history of the world, and therefore India has to play its role, right? And when it says that, when the world says that it is Indian Ocean, that means it belongs to us. We have the power to control. We have to have the power to control. So therefore, we have to have a sea denial navy, right? We can't have a coastal navy or just what is that known as? We can't limit our boundaries to just few hundred kilometers, right? Now we have to have sea control power over the Indian Ocean, right? Yeah, if you see Cholas, they had the complete control over the Malacca Strait, right? No, they were good seafarers. In fact, the Indian Navy started. If you look at, India is a maritime country, right? So we are born with a navy, right? So therefore, we have the potential. Only thing is that we need the support of the scientists, students, and professors to contribute more towards the maritime research in the development of submarines, ships, and the aircraft. In fact, whenever I meet an IIT professor, I keep asking, tell me your IIT, how much it has contributed for the development of sonars, aircraft, or the ships, right now. Now, why only the Navy? That is what another thing I want to say, that if you want to become military superpower, how to become? Which one to make a song? Air Force or the Navy or the Army? If you look at, Britishers had the brain. They felt that the earth has got 3/4 of water. If we rule the waters, then we can rule the world, because all the land masses are covered by waters. So if you cover the, if you rule these waters, then you can cover all the land masses. Land masses are the ones which are having resources which are easily exploitable, though the sea resources are also there, and we have the people inside, and therefore if we capture, we can capture the entire resources, right? You can capture the world. So therefore, it shows that that's how they capture the entire world. In fact, all the countries, if you see, they got freedom from England, right? Now you see all the small islands. If you see, there will be a bracket, British islands will be there. Why? Because islands are the strategic places where you can dump all your ammunition, ships, and aircraft, and have a far-reaching capability, right? Now, if you have to fight a war 10,000 kilometers away from a mainland, then you have to sail all the way. You have to have bigger ships, bigger submarines, and more weapons, right? Now, but if you have in between islands, then if you have those bases there, then what happens is your reachability is better. Your sea denial or sea control capability is better, right? Now, so that is the reason why islands are very important. That's the reason why we are spending a lot of money on this Andaman Nicobar Islands, and we want to develop the islands, and we are, having a tri-command service at Andaman Nicobar. And almost all the wars in the past have been amphibian wars, and the future wars are going to be again amphibian wars, and therefore they will be fought over the land, air, and the water, right? So therefore, I said very clearly now that India cannot remain away. India has to become military superpower, and the Navy has to be made very strong, because I told you very clearly that if you one who rules the waters will rule the world, right? Okay. How to become a superpower? Okay. Now, what are the problems that these are the things which we have to look at, right? Indigenous avionics systems. As of now, if you see, we have the capability to manufacture helicopters of small size, of low all-up weight, and small aircraft, but we don't have the wherewithal to develop the bigger aeroplanes, wide-bodied aeroplanes with all kinds of mission suits. That's what I said. Capability to build bigger aircraft with mission suits. That is integrated systems. Okay, of submarine detection, or ship detection, or maritime surveillance, or electronic warfare, and things like that, right? So we need to have bigger aircraft so that they can carry more fuel, more weapons, and have more endurance, right? Now, so then only you can have sea control over the vast Indian Ocean region or anywhere else, right? So therefore, we should have the capability to build bigger aircraft and capability to make better avionic systems. Still, we are buying from Israel and other countries, right? That we need to minimize. So when we are talking about Make in India, or whatever, the two things which Dr. Vasiki Madam mentioned, or the Prime Minister says that Make in India, or for that, what we have to do is we have to have more participation from the IITs, IISc, NITs, or the best of the institutions, or the students, the best of the students, right? Now, we have to think how much I have contributed. Every professor, every scientist, every engineer on this country should ask himself, what I have contributed, how much of percentage I have contributed towards improvement of the defense. Forget about, yes, not worked in the defense, no problem, no problem. He has earned so many degrees, so many qualifications, he has done so much of research. I want to ask everybody, how much of percentage you have contributed for the development of the military or the defense of the country. Ask yourself, please put in efforts, right? Now, orient towards that. This is the question I want to leave with all the students here. In fact, I keep asking everywhere, wherever I go, and I make everybody to think on this line that at least 1% if you have contributed in your entire lifetime, I'm telling you, you've done your job, right? Capability to build nuclear-powered aircraft, carriers, ships, and submarines. Yes, very important, because nuclear propulsion gives long endurance. The nuclear submarines can sail for 2-3 months together. They can dive deeper, okay, and they can do a lot of missions, right? So therefore, we have a problem here. We have got diesel submarines. We have just started making nuclear submarines, and of course, we have experimented with Russians and nuclear submarines for the last 30 years. We have got the experience. We have got the wherewithal with respect to the human element. We can sail a submarine, nuclear submarine. We can fight a war, but we are unable to produce more nuclear submarines. The second one is on the roll, right? So therefore, that is another area we need to concentrate. And as of now, we don't have a nuclear propelled ship. We don't have a nuclear propelled aircraft carrier. we need to have at least 3 aircraft carriers minimum. Luxury will be 4, right? No, but as of now we don't have any, right? We have just got one, and the one is about to roll out, or but it will have its sea trials in another 1 or 2 years. So therefore, nuclear-powered aircraft carriers and ships and submarines, the focus area, and a lot of researchers have to go on, and people have to participate, right? No, if we are looking at only money, if we are only looking at publishing papers to make money, right? We have to be feeling ashamed. I'm telling you, okay? No, we have to be ashamed. We have to ask this question as to what have I contributed towards this for the defense of the country, right? No, towards these questions that I'm posing in front. Please think, right? Unmanned aerial vehicles. We have unmanned aerial vehicles. We are only carrying out electronic intelligence warfare, but we are not carrying active. It is only a passive warfare that we are doing. We are not doing active warfare with weaponized and more all-up weight, more endurance UAVs, right? So therefore, why I thought this, I'll emphasize this, because there's a lot of fantasy going on in the community, in the students community, in the colleges are making UAVs for various purposes, right? We need to focus on, our country needs to focus on making bigger UAVs which can carry more of all-up weight and more sensors, more processing systems, and weaponized UAVs, right now. So these are the challenges that are in the front, and I request all the students to go on this. And there are few areas I thought I'll just discuss with you where you can focus, because I can see a lot of science people, material science students here and professors here. I thought I'll just leave a thought process here on this note. Battery, it is a very important area where we are very, very weak, okay, no? All over the world, in fact. Every gadget or system needs power. High electrical power of long endurance is what is required. Submarines, they require batteries. We cannot have a submarine without a battery, even if it is a nuclear-powered submarine, right? So therefore, what happens is, and it can't occupy more space. Because space is a premium on board a submarine, that we have to have very less space for the battery. So that means the size of the battery has to be small, and the charging time has to be very, very less. Maintenance should be very, very less, and it should power all the systems on board a nuclear submarine. Therefore, it's a challenge. Therefore, it's a challenge, right? So we need to think about developing materials which can store power, and it can deliver for longer duration for high power, right? Similarly, the UAVs, you cannot have an engine or a heavy machinery on board a UAV, because the all of it will get limited, because our focus is to carry more avionics, more weapons, right now. So therefore, the basic things that are required for the UAV to fly in the air for a longer duration has to be of less weight, of less size, right? So that is the area we need to look at. And this battery is very, very important, because sonobuoys, we drop sonobuoys to from the aeroplanes to track the submarines, detect and destroy, and sonobuoys carry the batteries. Now sonobuoys, when we buy from abroad, we buy in hundreds, and what happens is the minimum order quantity problem is there. You cannot just buy 1 or 2. We have to buy in thousands or hundreds. Okay, and when you take, what happens, every battery has got a life, you all know. And if it is only some 3 months and 6 months, in the storage period itself, the battery life goes, right? So we are manufacturing indigenous battery, right? This indigenous battery we fit it, but the battery lifetime again is very less. We have got only about 2 to 3 hours of lifetime, whereas for carrying out a anti-submarine warfare exercise, we need to have at least about 4 to 8 hours of battery life. So these sonobuoys, what happens is, during their time at sea, when you have dropped, when it is transmitting the submarine signals itself, it is switching off and going down, and therefore we are losing a lot of money on exercises, right? When we go for an exercise tracking a submarine, etc., we put so many sonobuoys. Each sonobuoy costs so heavily, so much money, right? And another one is sonar locator beacon. Any aircraft that goes inside the water, whether it is a Malaysian airliner which went, or the military aircraft that goes inside the water, how to locate where is it has gone, right? The only source is the sonar locator beacon. It has got a beacon. When the aircraft goes inside the seawater activated battery, it starts working and it starts emitting a beacon, okay, a signal. So the the search aircraft, when it is flying over it, right now, it receives the beacon, and that's how it locates. Now the problem is that when the aircraft dives inside, from that moment it starts sending the beacons. Now by the time the ATC notices that the aircraft communication is lost, or the beep has gone, and it intimates the ground, other people and the country, and you ask the support, and you launch your ships or submarine or the aircraft, it takes about 3 to 4 hours or more than that. By the time this battery life is gone, and it switches off. Therefore, there is no clue of where that aircraft has gone. So imagine thousands of crores, people inside, it has gone inside. The small battery can scoop everything, right? So therefore, battery plays very, very important role. And that is where we are very bad. We are not so good, right? Though we have, advanced batteries and things like that, but still, it's a long way to go, in my opinion, right? So lead acid, it is a lot problem, and lithium-ion batteries we have, but then not sufficient. Today, that whatever we have, right now. So you can see here, this is a nuclear submarine. You can see so much of space it occupies. This is a battery compartment, okay? And this is not affordable. No way, okay? We can, we are losing so much of space where we can have so many torpedoes, right now. So therefore, this is a focus area where a lot of research is required. And these are the sonobuoys. You can see the battery will be at the bottom, because when the sonobuoy falls vertically with the parachute, this part, half of it goes inside the water, half of it is outside, and the underwater side, it emits the sound waves, and it gets reflected from the submarines, and that's how it detects. And the antenna which is on top, it transmits to the aircraft and receives, and we identify the submarine, right? So this is the UAV you can see. So we need to have solar powered and things like that. Okay, submarine communication, another big major issue. We are unable to communicate to the submarines right now. Even today, all the best of the countries, they are unable to have VLF stations because they are very costly. US and Russia, they are unable to. Even Australia has closed down, and India has one near the Ramnad. We have our own submarine communication. It costs very heavily because when you go for low frequency, the lambda is very high, of several thousand kilometers, and therefore the antenna size and the power that is required to maintain, keep the antenna and the system alive is, too much, right? And of course, the Russians have done it. They have established at Murmansk, where I have been once at 72-degree latitude in Arctic Circle for buying the aeroplanes I had gone. In fact, I was the first foreigner to visit Murmansk air base of Russia, right now. And so I've seen myself these places, and it is impossible. Of course, with that, even the Antarctic has received from Arctic Circle the transmission that they have done. The Antarctic has received, stations have received, but then it has got its own problem of endangering human life and the animal life, and therefore, the questions are there as to should we go for this? How to communicate with the submarines? Now, the submarines are inside water. How to do it? And we have got a biggest problem of the seawater, which has got salinity and temperature gradients, right now. So therefore, sonars have got always problem, and so therefore, the best of the sonars are unable to detect, and still the detection, we don't have a system which can detect the algorithms. We don't have algorithms which can detect the submarines on their own. Still, we are dependent on the experience of the operator. Only through the sound that he listens, he can experience, he can point out that the signal belongs to the submarine. Otherwise, lot of clutter and so many things are there that we are unable to detect submarines because, so it is a challenge. It is a challenge for the entire world, right? Submarine communication is a biggest challenge, right? And with respect to submarines, the diving is a problem. We are only up to go up to 500 meters. Okay, so it is shallow waters again that it's a problem. So we should be able to dive. So metallurgy is important. Metallurgy, there is a problem. And even the propellers, you must have heard about cavitation, all the physics students, right? So that affects the propeller, and it creates pressure waves and sound waves, which we can capture and detect the submarines. Okay, so we need to have submarines and propulsion systems which will not produce any pressure waves or sound waves inside the water. Again, a challenge, right? So with all these challenges, it's a 110 now. I've got very less, therefore, I'm finishing my speech with this. And of course, there are so many problems, superconductivity, all of you know, at room temperature, still it's not available. When I studied at PSG Tech in 81, my professors, you all may be knowing, Professor Shashidharan Nair, Professor Jayakumar, who is there at the head of the Department of Physics at PSG Tech. So they all used to tell me that superconductivity is a challenge in 81, that if you develop superconductivity at room temperature, you can do it, but still, yes, nobody has found out yet. And replacement for silicon, of course, silicon is abundantly available, but it's a technological challenge to, develop a exact replacement for silicon. You all know because of the crystal structure and all that you must have studied, right? No, ranges of sonar is still a problem. Okay, we are unable to have good ranges of sonar, right? So, with respect to underwater, warfare, all the countries are very weak. Okay, so there is a lot of, in fact, you all must be proud that our own Indian, our own Tamilian, South Indian, Okay, no? Commodore Paul Raj, who was in the Navy, he is regarded as probably as the father of Indian sonars. In fact, he was the one who developed, hull-mounted sonars, various, varying depth sonars, etc. When in the 80s, and with his research, Indian Navy went by leaps and bounds, and immediately he was picked up by Stanford University. Moment he retired from the Navy as a Commodore, and he became a professor there. And if you type his name, you will find a lot of researches and outcomes. He has been awarded the highest honor by the US government, right? So it's a proud moment for us that he is the one who developed the sonars for the Indian Navy. Okay, no? And if the sonars are weak, you can't fight a war underwater or over the water, because the enemy's torpedoes can easily sink our ships and submarines, right? Right. So with this, I complete my speech. I hope it was useful. Though I've got less time of 20 minutes, I think I've driven the crux of what is required in this country today with respect to the naval warfare is concerned. And I also emphasized how important is Navy for this country, right? So I hope I'm able to send all of you for lunch so that 1:30 you can go for the exam. Yeah.
Thank you, sir.
Yes, ma'am.
Any questions from the participant side?
Okay, I'll just tell you something that I heard. Wherever I go, I generally give my telephone number, mobile number. You can note down, you can have a discussion with me anytime with respect to science, engineering, management, education, whatever are psychological problems that you have from birth to death, cradle to grave and womb to tomb. There is no problem. 24/7 365. I'm available right now for any kind of counseling. And so my number is 854-727-4749. I'll just type it here at the window. You can see 854-727-4749. Yeah. Okay. You can give it to your students. You can do your research scholars. Everybody. There is no issue. Absolutely, I'm available, and I can take speeches or lectures or sometime back, 1 year, I think, visited the as the for one of the functions. I have that vivid memories and good feelings. Yeah, have a good day. Bye.
Thank you, sir. Now I request R.T. Narendra Kannan, sir, Organizing Secretary, Assistant Professor in Department of Biochemistry, Kongunadu Arts and Science College, Coimbatore, to share the word with the participants. Please, sir.
Thank you very much, ma'am. Very good afternoon to one and all. I think it's too late for us. This is my duty to say a vote of thanks to everyone, those who participated in this program. First of all, I thank our Secretary and Director and Convener Coimbatore ISCA Chapter, Dr. C.A. Vasuki ma'am, for her constant support and encouragement throughout our endeavor. Secondly, I thank Dr. Vijayasimi Saxena ma'am. She's a General President in ISCA Kolkata for guidance programs, and I thank Bharatiya University Professor Dr. P. Kaliraj sir for readily accepting to give an inaugural address for this program. Thank you very much, sir. And I express my thanks to our Professor Dr. R.B. Subramaniam, BRD School of Biosciences, Sardar Patel University, Gujarat, and he spoke about strobilin and their role in plant defense development. And it was eye-opening, the new triterpenes, which is helpful for our further research in botany faculty members as well as botany scholars. And secondly, I thank Professor Dr. R. Karvembu sir, who is a professor Department of Chemistry, NIT Trichy, and he was talked about nanomaterials in catalysis, and he has given many input about the reactions like ruthenium and many other metals, which is very which we are not much familiar on the reactions, catalysis reactions. Thank you very much, sir. And I express my thanks to the Dr. N. Ayadurai, Principal Scientist, Associate Professor, ACSR, CLRI, Chennai, and here with his biological note, because we have started from botany and again turned to chemistry, then reached to biological sciences, and his talk about a green fluorescence protein, Nobel Prize winner for a discovery. And it was very wonderful, like a story. It was like a biological story. And finally, Dr. Professor Ramasubramaniam, Chief Administrative Officer, IIM Kashipur, Uttarakhand, and he was eye-opening that is a new, one of the best examples that India is a very old tradition, like only one ocean, Indian Ocean, in the name of the country. Rest of that, the region names. He talked about technology innovations in marine warfare. Thank you very much, sir. And I thank my colleagues and my senior Dr. Dhanam ma'am, Associate Professor and Head Department of Physics, for her support, and Dr. Sangeeta, Dr. Kartika, Dr. Rekha, and those who are behind the screen and organizing all these things from the beginning, arranging the webinar, and Dr. Rekha and Dr. Kartika and Dr. Velmurugan, who are supporting us in the behind the screen, Dr. C. Shobana also. And I thank each and every one, the participants from the morning 9:30 to till now. And we thank you one and all present here. And please fill the feedback feedback form which is posting in the chat box. And we will be sending you certificates soon. Thank you very much. Thank you once again to one and all present here. Thank you.